Good morning. The Nestle Nutrition Institute workshop number 94, the day number 3, and we still have people in the room. This is impressive. This is impressive, and it means that the topic we are discussing, which is milk mucosal immunity and the microbiome impact on the neonate, is really fascinating topic which attracts a lot of attention of people around the world. And today I have a great pleasure to introduce a chair of the third session, Professor Bo Lonnerdahl. His current position is Distinguished Professor Emeritus of Nutrition and Internal Medicine. He has a lot of achievements. The major ones are the Borden Award, American Institute of Nutrition, International Award for Modern Nutrition, Switzerland, Macy Georgi Award in International Society of Human Milk and Research, and Gabriel Bertrand Prize and Medal. And current interest, guess what? It's infant nutrition, bioactive components and milk, and iron metabolism. Session which Bo Lonnerdahl will be chairing today is about protective factors in human milk. And the great group of speakers will discover you what milk means in the terms of protection. Bo, please.
Thank you very much, Natalia, and good morning. I'm really pleased to be chairing this third day of our symposium, and I feel honored to have put together a program together with my co-chairs, Ray Ogran, Alan Walker, and we will today discuss what you have seen popping up now and then during the first days when we talked about the immunology of human milk, we talked about the microbiome of human milk. You have seen that we also, in breast milk, have some soluble factors doing something. And today we're going to discuss what these other things that are soluble are doing to various things in the breastfed infant. And we will start with the first speaker, who is Lars Bode, who I've known since he was a graduate student and I came to visit me at UC Davis. Lars is at our sister university, that is UC San Diego, University of California, San Diego, where he is the director of the Lars and Rosenqvist Foundation Mother Milk Infant Center for Research Excellence. And they're doing a lot of very exciting research on human milk right now. Lars has published extensively on oligosaccharides, and he has received several awards, prestigious awards for his research work. And it's a pleasure for me to introduce him to speak now about milk oligosaccharides, structure and function. Lars, please.
Good morning. Thank you very much for the invitation, for putting this program together, for NNI for hosting us. We heard about oligosaccharides quite a bit the first 2 days. The word popped up here and there, microbiome, prebiotic, oligosaccharides. So this morning we're going to really dive deep into what that means, human milk oligosaccharides, or short HMOs. Okay, a couple of words about disclosures, where the funding is coming from, and then we go right in. And I start with a statement that we've seen a few times in previous presentations in the first 2 days. And this is from the editorial commentary in the Lancet Breastfeeding Series 2016 that said that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion. And for us, the question is, why is that? How does it work? What is behind that statement? What is in human milk that makes it so powerful? So if we lift the curtain behind this statement here and look at what is in human milk, what are the macromolecules that we find in human milk? There's proteins, fat, lactose as the predominant milk sugar, and then there's oligosaccharides. 5 to 15 gram in mature milk, which often exceeds the amount of total protein, and in comparison to bovine milk is 100 to 1,000-fold more than we find in bovine milk. And since bovine milk is the basis for most infant formula, there's hardly any human milk oligosaccharides in infant formula, at least until recently. We've got to come back to that in a minute. So the talk is about structure and function. And I'm going to keep the structure part as short and uncomplicated as possible. We have to talk about it a little bit. So it's not as dramatic as this picture shows it. When we think of sugar, we think of something like this. When we think of sugar in the infant space, we think of something like this. That's fairly complex to me already, looking just at the structure. But when we talk about human milk oligosaccharides, which are complex sugars, that's not the same. So it's not the typical sugar that you think of that you associate something bad with. It's actually something good. And I'm going to show you throughout the hour what potential benefits there might be of these human milk oligosaccharides. So here's a fairly simple structure of a human milk oligosaccharide. You don't want to draw this out every time you talk about this. So we give every monosaccharide building block here a separate symbol, and then assemble them together depending on the structure. And really, there's only 5 building blocks. It's glucose and galactose, which we know forms lactose. And then in addition to that, we have N-acetylglucosamine, fucose, and sialic acid. So 5 building blocks, and depending on how you put them together, you get different oligosaccharides. So it's a little bit like Lego bricks. And you can imagine if you put Lego bricks together, you either get a car or you get a little plane. And you can imagine these two things do different things, right? A plane does different things than a car. And that's the same for the oligosaccharides as well. That's fairly simple structures, but structure determines function. And it can get fairly complex. That's my daughter getting the 1,200 pieces Lego set here for Christmas. It doesn't get that complex, so it's not that bad. Okay, so let's take a look at oligosaccharides and different structures. All human milk oligosaccharides have lactose at the reducing end. It's glucose and galactose. And then they get extended by disaccharide units in different linkages. They can be branched, further elongated. So you see already this gets fairly complex, either very long or branched. You can modify these structures with fucose in different linkages. If you add fucose on lactose, you get 2-fucosylactose, 2-FL, and 3-fucosylactose. Or if you add fucose at the growing chain, you get different oligosaccharides. And here an example for these 4 oligosaccharides. They have the same monosaccharide composition, but you put those Lego bricks together in different ways, you get different structures. And we think that these 4 oligosaccharides indeed have different functions. Gets a bit more complicated from there. You can also add sialic acid. Sialic acid introduces a negative charge through its carboxyl group. If you add sialic acid to lactose, you get 3 and 6-sial-lactose. And if you add it to the growing chain, you get different structural isomers. You can add more than just one sialic acid, like down here in this structure. And the important part really is that there's about 150, 200 different oligosaccharides identified so far, and every mom makes different ones. So it's almost like a thumbprint. Huge variation from mom to mom. I'm going to show you that in the next few slides. So what are we going to focus on? What's my lab focusing on? One is maternal drivers. What contributes to that variation in oligosaccharide composition between different moms? What's the benefits for infant health and development? And then can we learn from human milk and take some of that knowledge and apply that to develop new therapeutics outside the infant space for adults? So let's take a look at the maternal drivers first. If you look at this slide, this is data from the child cohort in Canada. It's about 1,200 subjects where we have oligosaccharide data for. And you see down on the bottom here, every bar is a different subject. And on the y-axis, you have in different colors the concentrations of different oligosaccharides. That looks very different, right? There is low amounts in these samples, very high amounts in these samples. And even if you look at relative abundance, so you set it all to 100%, the pattern looks very different from mom to mom. So like I said, this is like a thumbprint almost. You can take a look at oligosaccharide data slightly different. This is a principal component analysis in the 3-dimensional space. And you see here, every dot is a milk sample. There's about 10,000 milk samples in this plot. And the closer those dots are together, the more similar the oligosaccharide composition. The less or the further apart those dots, the less similar the oligosaccharide composition. And just by looking at this, So this is collected from 10,000 samples a set, approximately, and really from almost all over the world. So most of the samples come, of course, from North America, where we are located, but we have almost every continent covered and many countries included. So you can see already this distribution here in these two clusters, right and left. What is that? Is there two different types of moms? Why is there such a separation in the oligosaccharide composition? If you go through this and separate it by where the samples were collected and which cohort they come from, you see, for example, here the Canadian child cohort in red, and then in comparison, a rotavirus project collected in India in green, and there is some separation, like in top down and top and bottom, but that doesn't explain the left and right separation. If you separate them by a different characteristic, you can actually see, that explains that separation. What is that? That is what we call the secretors and the non-secretors. What does that mean? So that is genetically determined specific mutation in enzymes or in genes that encode enzymes that add fucose to your oligosaccharides. So it's shown here in the corner. There is an enzyme here called fucosyltransferase 2, or FUT2, that adds fucose specifically in this alpha-1,2 linkage. There's another enzyme, FUT3, fucosyltransferase 3, that adds fucose in this alpha-1,4, alpha-1,3 linkage, depending on the underlying structure. So it happens that there is people among us that have a specific SNP, or specific SNPs, so that's single nucleotide polymorphism, that inactivate this gene. So that enzyme is completely dead. It's a mutation that introduces a stop codon. And these moms, or humans in general, can no longer add fucose in this alpha-1,2 linkage here. They still have a FUT3, and that explains the difference between these two different clusters here. It's one base pair. One base pair in the genome that changes the entire composition of human milk oligosaccharides. It's not just one oligosaccharide that's now missing or in a low or high abundance. The entire oligosaccharide profile is completely different. One single nucleotide polymorphism. So the secretors here have an active FUT2, and the non-secretors have an inactive FUT2. And you can map this out by different cohorts. This is, again, data from the child cohort. In red, you see the homozygous SNPs. Orange is the heterozygous SNPs, and white is the wild-type, two wild-type alleles. You see how that clusters out. So that's your secretor and non-secretor. You can do the same thing with FUT3. So that's the fucosyltransferase 3. And you can see there's clustering as well. This time you have the dark spots, the homozygous SNPs, down here in the bottom, orange somewhere in the middle, and white on the top. So you can draw a line like this. So eventually you can cluster this in secretor positive, Lewis positive in this quadrant here. So that's active FUT2, inactive FUT2 and active FUT3. And you can go through the different quadrants and you see how genetics really determine the oligosaccharide composition on a global scale. This is not just one oligosaccharide. So we knew about FUT2 and FUT3 already, but we can take this a step further now and look through different oligosaccharides and how they're associated with different enzymes, and then try to understand which enzymes really are participating in the synthesis of human milk oligosaccharides, which we don't know yet. There's still an entire textbook page in biochemistry that needs to be written. So here, for example, we have a sialtransferase ST6gal1 that you don't really see contributes to any of the clustering here, but you see there is individual oligosaccharides that are strongly associated with SNPs in this specific gene. And here specifically you see 6-sialactose, which this enzyme here catalyzes the reaction of adding sialic acid in a 2,6 linkage here. And you see it's not a loss of 6-sialactose, it's actually an increase of 6-sialactose when you have that SNP. So there's a gain of function SNP. And that does not relate to 3SL. It relates to a similar oligosaccharide, that's the extended structure, LSTC, but not LSTB. So there's very clear associations between different SNPs and different genes and oligosaccharide synthesis. Genetics play a major role in oligosaccharide composition. So now we're using GWAS, so genome-wide association studies, transcriptomics, HMomics, and then use in silico pathway modeling and eventually in vitro target validation to unravel the pathway of human milk biosynthesis. Again, it's not known how most of these oligosaccharides are synthesized in the human mammary gland. And that changes depending on where you are on the planet. For example, if you look down here in Peru, or really in South America in general, most moms are secretors and have an active feud too. 98%, 95% in other countries. That's different on the African continent. You find 20, 25, 35, up to 40% of the moms that are non-secretors. They don't make these specific oligosaccharides. They make up other oligosaccharides, but their oligosaccharide profile is completely different. And that really makes you wonder, what do oligosaccharides do to the infant, to the mom, in different environments, in different parts of the world? Oligosaccharide composition changes over the course of lactation. You see, for example, 3-sial lactose goes up in concentration, 6-sial lactose goes down, DSLNT goes up, and you can go through multiple different oligosaccharides, and you go through multiple different cohorts, and you find the same trends in different cohorts. It also seems to change depending on the season. So we found in the Canadian cohort, and others have found that in a cohort in Gambia before, that there is specific oligosaccharides that change concentration depending on where you are in the season. It can be very extreme, dry season and wet season in Africa, but it also happens in Canada. So we don't really know why that is and what contributes to that, but there seems to be seasonal variations as well. We've also seen that oligosaccharide total amount is different whether mom had already a baby or not. So the more infants were already here, the higher the oligosaccharide concentration. That is not depending on age, so maternal age is not associated with oligosaccharide composition. And mode of delivery isn't either. It doesn't matter whether you deliver vaginally or by C-section. For the oligosaccharides, that doesn't seem to make a difference. So that's the genetics, and I think there's a lot of questions that we need to answer in that part still. But there is many, many more other questions when it comes to maternal drivers. For example, what about diet and exercise? Can we manipulate oligosaccharide composition that way? We don't know. We have some data here from a mouse model. Mice make oligosaccharides, 2 to be precise, mostly 3-SL, a little bit of 6-SL. And mice are much easier to work with when it comes to changing diet and putting them on an exercise regime. So you can measure 3-sial lactose concentrations in mouse milk and then put the mouse on a high-fat diet. And you see 3-sial lactose concentration goes down. You can exercise the mouse, and the 3-sial lactose concentration goes up. And you can do the cruel experiment to put a mouse on a high-fat diet and then exercise it. That's how I felt this morning, really. And you see that those two effects even each other out. Now you're back to almost normal to where you were before. Of course, this is mice. We don't know if this happens in humans. We have some data from a collaboration at Baylor, also from a collaboration in Arkansas, that points in the same direction. Sialated oligosaccharides seem to respond to high-fat diet, but also to exercise. So we're working on that right now to confirm that, but it seems to hold up. Then we have some data that showed that using probiotics during pregnancy changes the oligosaccharide composition during lactation. What about maternal health? Gestational diabetes, chronic inflammatory diseases, how does that affect oligosaccharide composition, milk composition in general, really? We don't have a lot of data around that. How about using medication? How does that change oligosaccharide composition? No data whatsoever. So lots of question marks still when it comes to maternal drivers. Why is that so important? Why do we need to know what changes oligosaccharide composition? Well, if we identify that there is specific oligosaccharides or specific oligosaccharide profiles that are potentially beneficial for the infant or for mom, then we want to know how that actually works. How can we manipulate that? Can we put mom on a specific diet or on a specific supplement to enrich for specific oligosaccharides that we identify to be beneficial? So in the infant health and development space, before we get to that, we need to know what happens to the oligosaccharides when baby ingests them, right? So oligosaccharides go in, they're resistant to the low pH in the stomach and to the pancreatic and brush border enzymes. About 1% gets absorbed in the small intestine, and the rest either gets degraded by bacteria in the colon or appear intact, excreted in the feces. And you see this huge variation here, 10 to 80% excreted. That's saying it's either everything or it's nothing. It's completely meaningless if you think about it, but really it depends on the stage of lactation. Very early on, you find a lot of oligosaccharides that come out intact. Then the microbiome somehow establishes and learns how to deal with these oligosaccharides. And then you find a lot less oligosaccharides coming out intact because now your bacteria is starting to metabolize them. And then towards weaning, when you introduce other foods, then you find more intact oligosaccharides, but also other glycans popping out in the feces. So it depends a little bit on where you are. That's why there's such a wide range here. So what do these oligosaccharides do? I'll give you this one cartoon here and go through this quickly. We always talk about human milk oligosaccharides as being prebiotic, right? So that's the food for bugs. And that might be true, and there might be a lot to it. And we started what we call Mommy Biome, which is a combination of the Center for Microbiome Innovation led by Rob Knight in San Diego and our Mommy Core. So we really try to understand how human milk oligosaccharides and other milk components impact the microbiome. But we think there is so much more to it. We think HMOs are way more than just food for bugs. We know that they're antimicrobials. So they directly act on the bad guys and keep them in check. We know they're anti-adhesives. I'm going to show you a slide on that in a minute. They directly modulate epithelial cells. And they also impact the immune system, either locally in the gut or on a systemic level. Remember, 1% is absorbed. Sounds like it's not a lot, right? 1% is neglectable. But 15 grams going in per liter, and this is every 2 to 3 hours. The kinetics tell us that there is decent concentrations of oligosaccharides in the infant blood. We find it in maternal circulation as well. So there's local effects in the gut, and then there's systemic effects. And we believe we are here. We've seen this iceberg before. We think we're just really now understanding what these oligosaccharides do, where they go, what target organs there are, and what mechanisms are behind this. So there is a lot to discover in this space still. Let's talk a little bit about anti-adhesives, and that's going to be my first story on infant health. What does this actually mean, anti-adhesives? So we have this pathogen here that approaches the intestinal epithelial cell, and to be able to hold on in this very dynamic milieu, they need to somehow attach to epithelial cells. And you can imagine if you're a bacterium, you're swimming around in the intestine, lots of movements, you want to hold on to something, because otherwise you're out pretty quickly. So the way they hold on is by interactions between bacterial lectins, so that's glycan-binding proteins, and glycans, sugar molecules, on the surface of your epithelial cells, the glycocalyx. So it's a thick layer of glycoprotein and glycolipid. And that is important for bacteria to be able to proliferate, and in some cases to invade and cause disease. Now, if we go one step back and look at the same diagram here, but now in the presence of human milk oligosaccharides that really come in constantly in very high concentrations. And it turns out that these human milk oligosaccharides resemble some of the epitopes that we find on the glycocalyx. They serve as soluble decoy receptors, bind to the bacteria. Bacteria can no longer bind to your epithelial cells, and they get flushed out. So that's why we believe that these anti-adhesive effects of oligosaccharides protect us from some of the infectious diseases. There's one example here that we've done with Lars Ekman at UCSD, who sits right next to me in the office, funded by the Gates Foundation, where we specifically look at EPEC attachment. So that's enteropathogenic E. coli. And we found in tissue culture that in the presence of human milk oligosaccharides, EPEC attaches less to epithelial cells. Attachment is not a very good measurement for disease potential. EPEC specifically causes these lesions on epithelial cells that you highlight here in light green. We can measure those lesions. So that's the damage that EPEC is doing on your epithelial cells. And in the presence of human milk oligosaccharides, there's significantly less lesions on epithelial cells. This is done in tissue culture. We're now repeating this in organoids as well, human organoids. And also we've shown that this works in neonatal mice. So you can add oligosaccharides to the diet, and EPEC colonization drops significantly. So that's one example, and you see here diarrhea is a major problem, 2,200 deaths almost per day, every single day, children under the age of 5. EPEC is not the most common pathogen for that. Rotavirus is one of the major causes for that. So we looked at rotavirus infection as well. We worked with Sashi Ramani here at Baylor in the Mary Estes group, and specifically at a rotavirus called G10P11, which is the spike protein that sits on this specific rotavirus that is found in areas in India, specifically in the neonatal age, so the first 4 to 5 weeks of life. And we thought, well, anti-adhesive must work here too. Glycans are involved in the binding. Let's test it out and show it, get it published and out. So Sashi grew epithelial cells, added the virus, looked at infectivity, and to our surprise, we found that there is specific oligosaccharides that increase the infectivity of this specific rotavirus strain. And we thought, oh, this can't be right. This assay must be wrong, because human milk is supposed to protect us. So how can this be right? We must be doing something wrong. So we repeated this a few times, different cells, different conditions, and we found the same results. We wanted to confirm this and went to India and looked specifically at the moms and the infants there, collected milk samples, analyzed them for oligosaccharides, and looked which infant developed or was infected with rotavirus and who had symptomatic and asymptomatic infections. And is there a difference in the oligosaccharides? And we found there is a difference in the oligosaccharides. This is looking at it more on a global scale here. This is principal component analysis. And you see already in red is the infants that are infected and show symptoms, so the milk oligosaccharide composition in corresponding moms, and they cluster slightly different. And then if we look at individual oligosaccharides, we find the same oligosaccharides that we found to increase infectivity in the in vitro assays were higher in the milk of moms where the infant got infected and had symptoms. So there was a clear overlap between the in vitro data and what we found in the mother-infant cohort here in India. So that was a surprise, to say the least. Is it possible that there are pathogens now that are getting ahead in the arms race and are trying to take advantage of human milk components? If that's the case, we need to know that. And can we use that knowledge then to get ahead, to develop better vaccination strategies? So it turns out Rotovac, which is the virus that is the vaccine that is used in that area, is a live attenuated strain, and we can increase the infectivity of Rotovac as well with the same oligosaccharides that showed an increased infectivity in the regular strain that we find. So can we add oligosaccharides to vaccination strategies and make vaccinations more efficient? So to summarize this part, specific HMOs are associated with increased rotavirus infection. And again, this is a specific rotavirus strain. This is not for all rotaviruses. And this is an opportunity to improve vaccination success. Another example is on growth. Are oligosaccharides related to infant growth? We had shown that with Michael Goran's group at USC, that there are specific oligosaccharides that somehow associate in cohorts with infant growth, fat mass, and all those parameters. And here we work with Kim Michelson's group in Copenhagen. He has this very unique cohort of exclusively breastfed infants with excessive weight gain. So if you look at the characteristics of these, there is a high weight gain group, and there is the weight for age z-score over 2 standard deviations. So these are gaining weight extremely quickly. And then the normal weight group here is between -1 and +1 standard deviations. And we wanted to know, is there a difference in the oligosaccharide composition between these two different groups? Can oligosaccharides explain this immense excessive weight gain in these infants? So we looked at total oligosaccharides. There was no significant difference in total oligosaccharides between the two different groups. We looked at HMO-bound fucose as a total measurements of fucosylation. There was a significant difference, more fucose in the high weight group. We also looked at HMO-bound silation, and there was no significant difference, although there was a trend in lower silation in the high weight group. And then we looked at specific individual oligosaccharides, and we found that specifically 2FL, 2-fucosyl-lactose, was significantly higher in the high weight group. Neo-LNT was significantly lower in the high weight group. So we have different oligosaccharides that seem to be at least associated in different ways, in this case with high weight gain. Now this is an exploratory study. It was only 30 mother-infant pairs, very small cohort, although we got significant results. And we wanted to confirm this, or let's go, let me put this here first. So this is now on a linear scale, 2FL and Neo-LNT. And just to give you an idea where we are currently with adding these oligosaccharides to infant formula, here's your 1 milligram per ml threshold. So we're way below that, way below the ranges that we measure in this study here. And Neo-LNT is right in the middle. And so in case you were wondering, are we now adding 2FL? Our kids are going to get fat now. We are way out of that range. Right? So at this point, no reason to be concerned. So that was 30 mother-infant pairs. We worked then with Hanne Lackström and Dr. Radava, who spoke yesterday, now went to a Finnish cohort, the STEPS cohort. These are fairly healthy infants, now 5, 6 years old, and we had a chance to look at 800 milk samples from 800 mother and infants. And we found the same thing. And this is presented a little bit differently here. This is the first 5 years of life. Here is your height standard deviations. And you see that the highest quartile in 2FL concentration gives you a higher height. And the lowest quartile gives you a lower height. So the same that we found earlier, the more 2FL you have, in this case the higher you are, but this is the same for weight as well. And the less 2FL you have, the smaller you are. It looks the other way around for Neo-LNT, just like we've seen in the Danish cohort. And if you take the ratio of 2FL and Neo-LNT, you see that this holds up nicely, and it holds up until 5 years of age. This is significant until 5 years of age. So you can imagine, depending on what oligosaccharides you give and what the ratio between different oligosaccharides are, you get the tall Scandinavian, or you get the smaller German guy, that would be me, and there's a difference of half a standard deviation just by looking at the 2FL Neo-LNT ratio. I think this is quite remarkable. So again, specific HMOs are associated with infant growth, infant and childhood growth. We need to study causalities. Is this just an association, or is there really a causality behind this? How does this work? And this could be a great opportunity to improve growth when it's needed. Another example is in the premature space, just one slide, although this is work that now has covered 10, 12 years of the work that we're doing in the lab. But we try to summarize this in one slide here. Briefly, what is NAC and why is it important? 5% of all very low birth weight preterm infants develop this often deadly intestinal disorder. We know that preterm infants who receive human milk are at 6 to 10-fold lower risk to develop NAC. Is there a connection with HMOs? Can HMOs explain this difference? So we set out about 10, 12 years ago to test this hypothesis that human milk oligosaccharides are somehow related to the lower incidence of NAC. What we did is we isolate oligosaccharides from pooled human donor milk to get the entire collection of human milk oligosaccharides, test that in an in vivo model, in this case in a neonatal rat model. We found that there is an effect, and we wanted to know which structure is it. So we went in, used multidimensional chromatography to really narrow down which structure is responsible for the effect that we see. And we found that this is the oligosaccharide that was responsible for the protective effect. It's called DSLNT, disillyl lacto-N tetrose. Then we presented this work, and we got slashed a few times saying, well, this animal model is not really good when it comes to necrotizing enterocolitis. You know, this has nothing to do with NEC, really. We looked at the chemical space around DSLNT, so we did a few studies, worked with the chemists to make derivatives, and really found that DSLNT was the most important component to be effective here. But we wanted to get around that criticism that this is just an animal model. So what we did is we designed a mother-infant pair cohort where we recruited specifically mothers and their very low birth weight infants in North America, collected milk samples every second day of the milk that went into that infant on that day, analyzed the human milk oligosaccharide composition, and compared oligosaccharide composition between infants that developed NEC and infants that did not develop NEC. And we found, to our surprise, that the same oligosaccharide that was protective in the animal model was lower in the milk that was given to those infants that later developed NEC. So it was protective in the animal model. If it was in low concentrations in mom's milk, that infant would develop NEC. So it was a clear match. We've repeated this in a study in South Africa in the meantime and found the same oligosaccharide being associated. Here we can say, well, it's an association, it's not causality, but since we have the animal data as well, there's a nice overlap that now gives us more confidence that we can move forward to a clinical study. And it also gives us some confidence that the model is not that bad after all. So studying the mechanism with the compound will give us some insights how this really works. So this is an example in the premature space. One specific compound. How are we doing with time?
Fine. 37 minutes. That's fine.
Okay, good. Give 10 minutes maybe to talk about something completely different. I know this seminar is about the neonate, impact on the neonate, but we have very exciting data to show that what we learn from human milk oligosaccharides in the infant space, in the neonatal space, can be very valuable when we go in and develop new therapeutics for adults at the other end of the life spectrum. So when we talk about frailty, atherosclerosis, arthritis, those kinds of things, we think that oligosaccharides can be very powerful in that space as well. I'm going to show you some data on that. So this is about chronic inflammation. It all started around 2014 when I went on a hike with a friend. This friend here is Philip Gortz, who's also a professor at UCSD, this guy over here. And the hike is up to Potato Chip, as we call it, in San Diego. And this is the chip that you're sitting on here. My daughter takes that very literal here. And so Philip is working on macrophages, chronic macrophage inflammation. He said, Lars, we need to find a way to work together. I would love to test your oligosaccharides, throw it on my macrophages, and see what happens. So we did that. Again, isolate all oligosaccharides from human milk, and threw that on his macrophages, and we found, wow, it's a significant reduction in IL-6 and IL-1 beta when you stimulate the cells with LPS and then throw your oligosaccharides on it. We then used the same approach to identify specific oligosaccharides that are responsible for that effect. And we found that 3SL, 3-sial-lactose, is the oligosaccharide that drives most of these effects. We found that this works in a mouse cell line. It works in primary cells. It works in human cell line as well. And it works in human patients as well. And we've shown that this is not just working on the RNA level, it works on the protein level as well. So the secretion of these cytokines is significantly reduced. We then went in and did dose finding, IC50 studies. So we found the IC50 is around 20 microgram per ml, which is close to what we have in the infant blood. It's not an unreasonable concentration. And we wanted to know, what is the mechanism? How does this work? How is 3SL reducing these very prominent pro-inflammatory cytokines? And to make this story short, I'm going through the next slides quite quickly. I'm going to show you one summary in the end. So we wanted to know, what's the receptor? Is it somehow interacting directly with the TLR4 receptor for LPS? And we found that's not the case. So we did RNA sequencing. We found that we stimulate the cells with LPS. You get a bunch of genes up and down regulated. But it's not the same genes that you affect with 3SL when you add that to the mix. So there's only a few genes that are differentially expressed in the presence of 3SL. So very unlikely that there's a direct interaction on the receptor, otherwise you would shut down the entire program. Then it's a sialylated glycan, so we thought, well, maybe it's the Siglecs. And we have the Siglec experts, right, one floor down, Ajit Barki and Victor Nizet. So they had knockout mice already running around. We just needed the bone, get the macrophages isolated, and see if our compound still has an effect. And it turns out it still works. So knocking out those Siglecs is not having an effect. Siglets are not the receptors. So what is the receptor? And now it really becomes a fishing expedition, right? It could be anything. It's very difficult to narrow down glycan-protein interactions. The affinity is not very high, so to do pull-down experiments, mass spec, very difficult to do that with a glycan. So we went back to our RNA sequencing data here. And we looked which genes are actually downregulated with 3SL. And not a surprise, it's the genes that are associated with inflammatory response, cytokines, cytokine activation. Which genes are upregulated, and we found, oh, that's interesting. There is a bunch of lipid metabolism involved here. And we found that there is all these genes that are associated with what we call an accelerated TLR4 resolution. So when you switch on a macrophage, you need to switch it off. That's the resolution phase. The faster you can switch a macrophage off, the less inflammation you have. So it turned out there was a paper published just a year before we looked into this from a guy at UCSD, Chris Glass' lab, that showed that TLR4 signaling switches on your inflammatory response here very early on. That's 1 to 6 hours. That's when all your cytokines are released. That's your active macrophage. But then it gets switched off by this pathway down here that includes a lot of lipid metabolism. And that happens in the 12 to 24-hour range. And it turns out that almost 90% of the genes that we saw induced by 3SL have something to do with this pattern here. So we went to Chris, asked him, can you help us with this? Can we throw our compound on it and then do ChIP-seq? So we did this experiment here, just to cut this very short now because it's very detailed, but just to show you that we're really going deep into this. So if you look at 2 hours, just LPS, you see all these NF-kappa B, all these different inflammatory pathways go up. But here you see the lipid metabolism coming up when you add 3SL to the mix. So let me skip this one here and go right to the model. Make this a little bit faster. So what we think is going to is happening, let me skip this one here as well. What we think what's happening, I'll skip this one here as well. That's what happens when you have too many slides. Okay, so here's the model. What happens when you add LPS or another stimulator? You switch on the inflammatory response and you get to this point here, right? Inflammation is switched on. This is your early phase of macrophage activation. While we still don't know what the receptor is, 3SL somehow interferes with this program here, shifts the macrophage from activation to resolution much faster. What happens in 24 hours now happens in 6 hours, and the macrophage gets switched off much faster. The lipid profile changes rapidly, much earlier these lipids pop up. We see the receptor for it that we can knock out. Now gives us that switch off. So the macrophage is switched off much faster. Now, why is that important? One of the diseases where chronic inflammation, macrophage inflammation, plays a significant role is rheumatoid arthritis. 1% of the world population has RA. That's about 1.3 million people in the US alone. And women are affected 3 times more than men. But they may also hold the secret on a potential therapeutic. So we collaborated here with Mary Pat-Kaur and Nunzio Bottini at UCSD and tested out our 3SL in a RA model, in a mouse model. We put 3-sialactose in the drinking water. First, to show you, we measure the paw thickness here throughout the time of disease. We induce the disease by giving it a collagen antibody. Paws swell up. You give it an LPS boost, and the paws get really inflamed. Now you add 3SL in the drinking water, and you see there's a significant difference in the paw swelling. A lot less paw swelling. And if you show this to someone who is in the RA field, will tell you, wow, this is what we see with our TNF-alpha inhibitors. This is very comparable. And not only that, at the end of the study, you see there is less inflammation, less bone erosion, less cartilage damage. So all those readouts that are typical for arthritis are significantly reduced in the presence of 3SL in the drinking water. Right? This is not an injection. This is just giving something in the drinking water. So we tried this out in the second model. This is a genetic model, and just to make this short, we find the same in 3SL reducing the paw thickness significantly. In the meantime, there's a group in South Korea that used a third independent model, also tried 3SL, and also found that 3SL significantly reduces arthritis. And that's about the same concentration. So here now we have data from an in vitro model, a cell model. We have data from an in vivo model, actually 3 independent models where we show that this compound works. Is there a human cohort where we can see if this really translates to human disease? Now, that's difficult to do because most of our infants don't develop arthritis, of course. But when we showed this data to Christina Chambers at UCSD, she said, well, that's interesting because we have so many women with RA that go in remission at the end of the first trimester, and then they flare up again towards the end of breastfeeding. It's like, well, that's interesting because it turns out that human milk oligosaccharides, specifically 3SL, pops up in maternal circulation at the end of the first trimester during pregnancy. So is there anything, is there a link between these two things? So what do you do if you want to do a cohort study? You go to Canada, you go and call Megan Azad and ask, did you ask your moms if they had arthritis? And it turns out that they collected data on arthritis. They asked moms, have you ever had RA? And if they said, no, we had never had that, we call that 0. That's our control group. Have you ever had RA, but you currently don't have RA? So that's our remission group. That's our group 1. And then, do you currently have RA? Still have RA while you're actually giving us a milk sample? That's our active group. And then we looked at the oligosaccharide composition, and we found, indeed, no difference in total oligosaccharides, but significantly more 3SL in the moms that went in remission. So a very nice correlation now between the in vitro data in tissue culture, mouse models, and coming in with a cohort like this where you show that moms that go in remission make more 3SL and produce that in the milk. So 3-sial lactose may be a potent therapeutic to treat and prevent arthritis. But there's other diseases that are related to chronic inflammation, chronic macrophage inflammation, and one is cardiovascular disease, the #1 killer in the world when it comes to annual death here. 18 million people die from cardiovascular disease. That's your heart attack and your stroke every single year. And macrophages play a huge role in that. That's your foam cells, right? That's the foam cells that suck up all your LDL, all your lipids, and then clog up your arteries. So to make this story short, again, working with Philip here, we put mice that had a LDLR knockout, so that's your receptor knockout, put it on a high-fat diet with and without 3SL, and then we looked at atherosclerosis assessment. No difference in food intake, no difference in weight gain, but a significant difference when it comes to atherosclerotic plaques in these animals on 3SL. And you can measure that in 3D. You can measure plaque size and necrotic course and all these things. And again, there's a significant difference in all those measures when the mice are on 3-sial lactose. So can we take a human milk component and apply that to patients that would potentially die from a heart attack or a stroke? A natural component that we give to babies every 2-3 hours anyway, potentially safe. This could be a real game changer. So summary here, 3-sial lactose may be a potent therapeutic to treat and prevent cardiovascular disease. That's just 2 diseases, really. RA, cardiovascular disease, I talked a little bit about diarrhea. There's so many other things, infectious superbugs, obesity, diabetes, IBD, breast cancer. There's always a link somehow to human milk and to breastfeeding. And we want to identify how this all works. And we call that our milk moonshot. We want to know what is in human milk, how this works, and how it could be protective for the infant, for mom, but also for adults that are at the very, very other end of the life spectrum. I keep you with that and take questions.
Dr. Mestecki first.
Carbohydrates in any situation can be, as you mentioned, inhibitors of adherence of microorganisms to receptors on epithelial cells. Surprisingly, unless I missed something, one of the important carbohydrates is mannose, high mannose type, oligosaccharides, and linked to asparagine residues. You don't have any mannose there.
No mannose.
So can you explain why there is no mannose, first of all? And second, sialic acid, alpha-2,3 or alpha-2,6? Because again, that makes a great difference with respect to recognition by influenza virus and so
Absolutely.
on, as inhibitor, that is.
Yeah. So no mannose in human milk oligosaccharides. Mannose is specific to your N-glycans. We don't find it in human milk oligosaccharides at all, which might give away a little bit how they're synthesized in the mammary gland.
But you find it on glycoproteins.
You find it on glycoproteins,
Oh,
you find it for sure in the milk, but you don't find it in human milk oligosaccharides. Yeah.
Okay.
And the other question was about the alpha-2,3, alpha-2,6 linked sialic acid. We find both.
Both.
Yep.
Okay.
And in different, interesting here is you have an alpha-2,6 linked sialic acid not only on the terminal galactoses but also internally on glutamac, and that is very unique to human milk oligosaccharides, really. Yeah.
Dr. Levchich first, and then Dr. Ogra. Carlos.
Congratulations, it really was a fantastic presentation and excellent work. I assume that the data that you presented of the higher height of children with the 2FL, that data is corrected for mid-parental height.
Is corrected for, sorry, I couldn't hear.
That the data, the results of the height is corrected, is adjusted for the mid-parental height.
For mom's height, you mean?
Well, yeah, because, I mean, it could be that the same way that genetics plays a role in determining a higher 2FL might determine that the parents are taller. And that's the child is tall not because of the 2FL, but because of the parental height.
Good point. I asked Samuli and I see him going this or that.
We corrected for maternal BMI and we corrected for birth measurements, but I don't think we corrected for maternal height, but that's an extremely important point.
Yeah, because the child's length at birth relates to maternal height, but at 4 years of age relates to mid-parental height. The father sometimes also plays a role.
I think that's a good point. Yeah.
Okay, Dr. Ogra.
These were really beautiful studies, and you presented them in such an elegant manner. Thank you very much.
Thank you.
I want to focus on the studies on the viruses, particularly rotavirus. Rotavirus, at least in the man, affects specific receptors on the terminally differentiated enterocytes. Mm-hmm. Do you know whether these oligosaccharides bind to the receptor on the enterocytes in any specific manner?
Mm-hmm. Yeah, we don't know exactly what the mechanism is behind it. It turns out, so the original hypothesis when we went in was to look for why is it that this specific rotavirus strain has a specific affinity to the neonate. Why is it that only in the first 4 to 6 weeks of life this specific rotavirus strain is found, and it doesn't infect infants or children at later age? So is there anything in the intestine? Are there glycan structures in the intestine that are different between the neonate and later on in life? So the receptor wouldn't find its binding partner anymore.
Because the disease in the rotavirus is very typically in the younger children, mm-hmm. but not in very, very young. In the newborn period, you don't see much disease with rotavirus.
This specific virus here, the G10P11, is specific for the first 4 to 6 weeks of life.
Is that
in
right?
that specific area in India.
But you don't know whether it binds to the receptor itself?
No.
Okay. The second question is, if it does alter the interaction of the virus and the epithelium in some way, do you see any changes in the induction of immune responses?
We haven't looked at that.
Or expression of disease?
We only looked at infectivity in the in vitro model, and then in the cohort we looked at whether there is infection and whether there is symptoms or not. Yeah.
The last question I had was related to not a virus but a bacteria like Clostridium. Mm-hmm. It's another good example. Clostridium is an infection which you see a lot of disease in adults and old people, but not in children. In the neonatal settings, they have a lot of colonization, but you don't see much clinical disease. Mm-hmm. Have you looked at any other bugs besides the ones you talked about in neonatal period?
Yeah, we've looked at a bunch of bugs. We looked, in addition to EPEC, we looked at Campylobacter, we looked at different E. coli, EACs, we looked at ETACs, we looked at Entamoeba lytica, the parasite, Giardia.
But did you look at Clostridium?
We didn't, no.
Which will be something very interesting to see, because that's another very unique setting in which this may be, you may have found a counterpart of steroids, for example. You can treat rheumatoid arthritis to viral infections and everything else. It would be fascinating. But it's an elegant study. Thank you.
Thank you. Yeah.
Dr. Hanish, and then on the third row over there.
Impressive talk. I liked it very much, and it helped me to use my impression that I'm misplaced at this meeting as a glyco person. So my question is, do we have any structural evidence on the higher mass glycans, whether they express blood group type 2 structures at all, except for the pentaoses, which are known to express type 2 structures?
Yes, so the higher molecular structures go down in concentration quite quickly.
Yeah.
We see that they may be top 15 or top 20 oligosaccharides are, at least by total concentration, the predominant players, and the moment you get larger, your concentration drops significantly. I'm not sure exactly what blood types you find on the different higher molecular oligosaccharides. Depends certainly also on the secretor, on the Lewis blood group status, right? I mean, that really drives that tremendously. But I would imagine that recaps on the larger structures as well.
Yeah. I was wondering, because we found only blood group H1 and Lewis A, B structures, so no Lewis Y, Lewis X, and all these other guys. Yeah. Okay.
Last question from the third row there.
Thank you for your nice deliberation of the very critical things which would help in the baby as well as in the future predictions for the chronic diseases. So my question, have you found any difference in the malnutrited mother and the well-nutrited mother in the difference of the oligosaccharides?
Say that again. Can you repeat that? Do you find differences in?
In malnutrited mother, as well as the well-nutrited mother, have you found any difference?
Yeah, so we haven't looked at that, but there is data that came out of UC Davis together with Jeff Gordon's group as well, where they specifically looked at moms. I believe it was in Bangladesh, but I think they also looked with Andrew Prentice in the Gambia, and there were differences in oligosaccharides, in oligosaccharide composition in moms that were malnourished. Yeah.
With that, I would like to thank Lars very much. Thank you. And the next speaker is Professor Dr. Franz Georg Hanisch from University of Köln. And he is the head of a research group focusing on molecular glycobiology, very fitting under these circumstances here. He's published heavily and has a very active research program. And today, I'm very happy that he will present on oligosaccharides and viral infections, something we heard about yesterday, and how milk oligosaccharides work versus algal fucan polysaccharides. Dr. Hanisch, please.
Thank you, Bo Lernadal, for this kind invitation and introduction to my work. Today, I would like to give you a broad and concise overview of our work on noroviruses in particular, so I will not speak on viruses in general. Noroviruses are still a human health challenge. They are a genus of the family of the Calciviridae. They are simple, non-enveloped viruses, which means that they are not carrying glycans on their surfaces. And they contain a single-stranded RNA genome, which is surrounded by a capsid that is composed mainly of one major capsid protein, the VP1 protein. It's accompanied by a smaller protein, the VP2 protein, which has just stabilizing functions. Noroviruses, together with rotaviruses, are the most frequent causes of virally induced acute gastroenteritis worldwide, and this holds true for developing and developed countries. So the numbers given here may be not actual, but it is estimated that outpatients, there are 7 0.7 million in industrialized countries and 0.5 million hospitalization in developed countries, and at least 9 million hospitalizations in developing countries, with nearly 200,000 deaths per year worldwide. And among children, we have annually about 50,000 deaths. Noroviruses are a genus which comprises 6 genogroups, G1 to G6, and only the genogroups 1, 2, and 4 infect humans. The most predominant strain which caused pandemic infections is G24, and in recent years, in 2015, a new strain emerged, which is G217. A couple of other genogroups are infecting other animals, for example, pigs and mice and dogs. And the simple reason why these strains are more or less specific for their species is that we have no real functional animal model for studying human infection in an animal. The only animal which can serve as an animal model is oyster, because oyster expresses also blood group-like structures similar to the human structures, and we can use this model to some extent, but not for studying infection. But it's just the binding of the virus to the gastrointestinal tract. Well, as I said, the most prevalent strain currently still worldwide is the G2/4 strain, in particular the strain which has emerged first in Australia, in Sydney, in 2012. And during the year of 2015, a new strain emerged, G217, in particular in China and Japan. These 2 strains, which are currently the most relevant strains in human infection, show characteristic age distributions. G2/4 particular is infecting largely younger children and elderly people, whereas the G17 shows a more broader spreading infection pattern. So it is claimed that G217 has gained high affinity for carbohydrates, and this may explain its stronger spreading efficiency. Now, the virus to start or initiate an infection needs to bind to an epithelium, and the epithelium is covered by highly glycosylated glycoproteins. One typical example of these glycoproteins is the mucin MAC1 or mucin 1, which is inserted into lipid rafts of the epithelial cells and presents hundreds of oligosaccharides, O-linked oligosaccharides. And these O-linked oligosaccharides start with the addition of a GalNAc to the amino acids serine and threonine, and these galactosamine residues can then be extended to a core 1 structure by addition of a galactose, and this core 1 structure then can be further elongated either by addition of sialic acid, which serves as a stop signal, so these structures are dead-end products which cannot be elongated anymore, or a branch is introduced by introduction of a glucanac residue at the position 6 of the GalNAc, and this glucanac residue is then extended to form so-called polylactosamine type structures, which can carry at their ends blood loop structures, which contain fucose and GalNAc and galactose. So these structures are also found on those mucins which are secreted. So in the gastrointestine, also in the stomach, we are confronted with these membrane-bound mucins, which are the target sites for the virus. And we see also secreted mucins which form viscoelastic gels on the surface of the epithelia and prevent a direct contact of viruses and bacteria, of course. So they also express the same structures principally, although each mucin which is secreted, for example, in the stomach, has an organ characteristic glycosylation. So one of the mucins which is expressed in the stomach is the MAC5AC, and another one is secreted by deep gastric glands. This is the MAC6, which shows a very characteristic and distinct glycosylation. This slide I show just to give you an overview over the structures which are relevant in the context of this presentation, derived from either type 1 structures, which is galactose linked to subterminal glucosamine, or type 2 structures where the linkage is 4 to the glucosamine. The blood group structures are derived from by either the secreted gene-dependent fucosyl transferase, which introduces fucose to the 2 position of the galactose, or by the Lewis-specific enzyme, which introduces the fucose to the subterminal glucosamine. The structures which are relevant in the context of milk oligosaccharides are shown more or less on the left-hand side, and this is why I asked Lars Bode whether he has any evidence on the formation of these type 2 structures, which can also be formed. Let me go on. Now we are searching for anti-infectiva based on natural compounds, and the natural compounds we first thought of are human milk oligosaccharides, and we asked the question whether these can be effective competitors of viral adhesion to epithelia. Now, the idea is quite simple. The virus is a multifunctional or multivalent lectin. So on its surface, it expresses multiple copies of the VP1 protein, and the protruding domains of these VP1 protein are forming a lectin-like domain, which is able to bind carbohydrates. And also on the other side, on the epithelium, we have a multifunctional glycoprotein. So this interaction to overcome will be a challenge for monovalent short-chain carbohydrates, but we thought we should have a look at the larger structures expressing more than one fucose, and to use these larger structures as potential decoys to inhibit this binding process. The VP1 protein, the structure is shown here on the right-hand side, is composed of 3 domains: the S domain, which is the structural core domain, and this is additionally by 2 further domains, the P1 and the P2 protruding domains, which form at the end this carbohydrate binding pocket. We're using this VP1 protein, which forms spontaneously to virus-like particles, and on the other hand, a dimer of the P1, P2 domains. This dimer was used in crystallography studies and co-crystallography studies with milk oligosaccharides, whereas the virus-like particle was used exclusively in solid-phase binding studies. The co-crystallization was performed with a series of milk oligosaccharides, either defined standard compounds or mixtures of undefined oligosaccharides, because our initial idea was to see whether complex oligosaccharides exceeding the size of decasaccharides, for example, could bind to the lectin of the P domain, and could in this way in the crystal be entrapped. And these crystals could then be analyzed by either X-ray analysis or by multi-mass spectrometry. So we could have been able to identify those structures which were preferentially entrapped in these co-crystals. Unfortunately, these larger structures never gave crystals, so we had to give up this project. And we could demonstrate for a couple of viruses, viral strains, in this case here for G210, the Vietnam strain, that only smaller sized oligosaccharides could be co-crystallized. And here, for example, 2-fucosylactose and 3-fucosylactose, although they have structurally nothing to do with each other, were able to crystallize within the pocket. On the other hand, other structures which have a structural relationship to the 2-fucosylactose, like the lacto-N neo-fucopentaose 1, did not co-crystallize. The genogroup G24 has evolved within 40 years. So in 1974, the Washington species, the Washington strain, which is shown here, this strain shows a strong binding to blood group A structures, whereas the later evolved Sydney strain from 2012 shows a diminished binding to blood group A, which means that within these 40 years of evolution, the strain has already changed dramatically. And here you see the many mutational changes which have occurred and which affect in some way the structural aspects of the binding pocket of the VP1 protein. And this results finally in the orientation, or in the different orientation of this loop-like structure here, in particular, and to different binding of the 2-fucosylactose shown here as an example. So already these few mutational changes here occurring during 40 years have dramatically changed the specificity of the binding groove. So the result of this mutational change is that the newly emerged strain from 2012 binds much better to blood group H structures and less to blood group A structures. Also, the newly emerged strain G217 shows an important mutation when compared with the first emergence in 2002 in Saitama in Japan. And this mutation refers to the replacement of a valine at 444 to a tyrosine. Lectin pockets are generally characterized by the phenomenon that they contain very often aromatic amino acid side chains, which are highly conserved. And we can speculate that this mutation here has made this binding pocket much more sugar-philic. Now, we in Cologne were interested in finding structures with a higher binding potency to the virus-like particle in the fraction of high-mass milk oligosaccharides, which were kindly provided by Clemens Kunz from Gießen. He provided us 42 fractions of high-mass glycans, which were characterized by 2 to 6 fucose residues per chain. Well, our strategy was based on an array which is composed of neoglycolipids derived from oligosaccharides. These oligosaccharides have reducing termini, and we can couple to these termini phosphatidylethanonamine, and these neoglycolipids are then embedded into a lipid layer and expose their carbohydrate chain to the aqueous phase. So we simulate in this fashion the situation on an epithelial surface to some extent. And in this model, which was developed by Tanfasi in the late 80s, we have initially tried to show that we can use this approach. In this case, we used the less relevant strain G210 which is more important in oyster infection. And we could demonstrate that one fraction of this complex array was most potent, and this is a fraction of milk oligosaccharides here, of large, high-mass milk oligosaccharides. Now, we did the same with the 42 fractions provided by Clemens Kunz, and we could demonstrate that not all fractions were active, most fractions were inactive at all, but a couple showed some significant binding activity. Now, to get insight into the structural requirements in case of the G2/4 binding, we use also a couple of standard compounds like lactofucopentaose-1 and the neo isoform, and lactofucopentaose-2, the Lewis A expressing pentaose. And this clearly showed that the virus-like particles could bind to both blood group H structures, but not to the Lewis A structure. So there is some degree of specificity in this binding. But in contradiction to other reports from other investigators, there is no discrimination between H1 and H2. There were reports from researchers working with saturation transfer difference, NMR, who could show that there is a strict discrimination of the virus particles between blood group H1 and H2, and also data from X-ray analyzers which said the same. But actually in this maybe more realistic experiment, these tiny differences in affinities were not or did not become visible at all. So the virus-like particles does not discriminate between these structures. Now, we isolated the most potent, most binding active subfractions of some of these fractions, and did mass spectrometry analysis, and could demonstrate that these highly active fractions were characterized by multiple fucosylation. Up to 4 fucoses were here found in this deca saccharide or nonasaccharide, and we could also gain structural information with concern to the type 1 and type 2 structures at the terminus of this branched structure. Now, what we saw on the level of the neoglycolipid arrays, we could reproduce with mucins isolated from gastric juice of individuals which had been defined with respect to their Lewis and secretor blood group status. And we could show that the virus-like particles bound preferentially to the Lewis B structure and to, in case of the Lewis negative material, to blood group H structures, but not or much less to Lewis A. Now we tried to inhibit the binding of these virus-like particles with a series of milk oligosaccharides. And we started with a 2-fucosylactose, which can be referred to as a reference, and with lactofucopentaose-1. Well, both showed inhibition at high concentration. So in case of the lactofucopentaose-1, which is actually or should be theoretically one of the best binders, we found IC50 values in the range of 50 millimolar or even more. We now have better data which are not presented here, and I can tell you that all higher mass oligosaccharides like lactofucopentaose-1 and the respective neo isoform both have these lousy affinities, obviously, in this system. 2-fucosylactose and 3-fucosylactose have IC50 values in the range of 15 to 25 millimolar. So obviously, these monovalent oligosaccharides are less potent in the blocking of viral binding, and this could be due to the valency of the lectin on the one side and on the valency of the oligosaccharides in case of the epithelial surface. So the question is, how can we compete with this multivalent components in this system? And this is an artificial approach, so to say. We thought we use a scaffold like alpha-cyclodextrin and couple to this cyclodextrin fucosyl residues simply by an acid reversion technology. And the fucose was shown by methylation analysis by GCMS to be linked exclusively to the 6th position of the glucose in this ring structure. And the fucose was linked alpha-glycosylically mostly on the basis of NMR analysis. So we could isolate at the end mono to hexa substituted derivatives and could test these as binding inhibitors, and we could see that with the increased degree of substitution, there was a tremendous increase in efficiency of blocking. So obviously the valency is the most important point, and you will agree with me that these structures shown here have nothing to do with the blood group structures in milk or on an epithelium. So it's just the fucose which is important, and this fucose should be presented in a multivalent fashion. And this led us to the question whether there could be other compounds in nature which could substitute for these blood group specific structures in milk. And we found these fucans or fucoidins, which are expressed in many marine invertebrates and in the cell walls of many brown algae, in particular in the fucalis and the laminarialis. These fucoidins are characterized by long chains where the core can be either completely composed of fucose only, or it can be repeating fucose and galactose, either 1,3 or 1,4 linked. Here is a structural model of the Fucus vesiculosus fucoidin, which is characterized by a high degree of sulfation. So every second fucose of this structure is sulfated. So these fucoidins are known for some time, and they have been shown to exert a multitude of bioactivities, among others anticoagulant and antithrombotic activity, anti-tumor and immunomodulatory activity, and so on. And among others also antiviral activity, for example in the poliovirus, adenovirus, and against HIV and cytomegalovirus and herpes simplex viruses. But I have to say all these effects have nothing to do with the binding competition, so the mechanism here is completely different, and it's totally dependent on sulfation of the fucoidin. Now we have tested various forms of fucoidin as a competitor of the viral binding to gastric mucins, and we could demonstrate that it is highly effective as a competitor either in the native form or in a desulfated and partially fragmented form. So you can see here the masses of a partially fragmented form which has been generated by solvolysis. And here you see a partial structure of those oligosaccharides shown here, which were highly active in the system. Well, interestingly, when we tried to entrap the oligosaccharides generated from fucoiden in a co-crystal with the P domains of the virus, we could demonstrate that only fucose shows up in the binding pocket. So this is exactly what we had expected. There is a strong effect, but this effect has nothing to do with the specificity. It's just a fucose which is linked. So it's a low affinity binding, but with a high avidity. This slide just shows the concentration dependency of the effect, which should be seen in a competitive system. And this slide may be interesting because it demonstrates that the viral particles are able to survive in some way even at lower pH conditions as they are given in the stomach. So down to pH 2.5, they are quite active, actively binding to gastric mucins. Currently, we are testing a variety of different preparations of fucoiden from different species, of course, which is not shown here on this slide. And we are comparing the pure stuff with a more purified stuff, because purification means that solvents have to be used, that oxidation processes are involved in all these things. And all these chemical treatments have influence on the activity. Now, this is one of my last slides. We are currently trying to establish oyster as a model for binding or accumulation of virus particles in the gastrointestinal tract of these animals. We could demonstrate that actually the strains G11, G24 are accumulating in this system, but interestingly not the G217 Kawasaki strain, although this has been claimed to have a higher affinity to blood group-like structures. But obviously the blood group-like structures recognized by this strain are not expressed in oyster. We have already initial data in this model and can tell you that there is a certain degree of inhibition exerted by 2-fucosylactose, the milk oligosaccharide, in this system. So principally, this model can work. Let me sum up what we have done. We have shown that the binding of GT4 VLPs to human gastric mucins is sugar dependent. The most important sugar in this event is the fucose in blood group H1 and Lewis B related human milk oligosaccharides. However, the affinity of this specific interaction, or at least the apparent affinity, is quite low in this system because of the multivalency of the interactors. The IC50 values are in the range of 20 to 60 millimolar for these tested milk oligosaccharides. We have demonstrated the valency, the importance of the valency effect in an artificial fucose dendrimer with these compounds, and have demonstrated that polyfucoses like fucoiden and fucoiden-derived oligofucoses are potent inhibitors of viral binding. The sulfation of fucoiden does not play any role, so the mechanism of this inhibition is completely different from other viral inhibition. Okay, I thank you, and I just would like to mention my collaborators, Horst Schoten and Wassily Morozov from Mannheim, who are doing these oyster experiments, Bernd Hansmann, who is responsible for the X-ray co-crystallography, Stefan Jennewein, who provided a couple of milk oligosaccharides. Ten Fazy was involved in the initial neoglycolipid array, Clemens Kunz for providing the milk oligosaccharides of high mass, and Dolores Diaz from the University of Cologne for performance of NMR work. Thank you for your attention.
Thank you very much. This paper is now open for discussion. Yes, Arnel?
Thank you very much for the presentation. I remember a paper that must be 15 years ago or so, where they studied the inhibitors in human milk of binding on norovirus and found that that was not the soluble oligosaccharides. The most inhibitory factors were actually two glycoproteins, and what they're having, that was mucins and bile salt stimulated lipase. And what they have in common is the tandem repeats that are O-glycosylated.
Yeah.
Do you have a comment on that?
Yeah, of course. Well, this is exactly in line with what I said before. So the glycoproteins, in particular the mucins, are the much better competitors because they are multivalent. And in particular, MAC1, which is one of the most prevalent mucins in human milk, on the milk globule membrane, it's the most important glycoprotein. It's also secreted and it's also shed from the membrane, and you find it in high amounts in milk, also in the whey of the milk. And we have done a lot of structural work in the late 80s on this mucin. And actually, this mucin would be the better player in this system, but not the monovalent oligosaccharides.
Well, I was actually thinking, is it the fact that they have tandem repeats that are O-glycosylated, so you have many different binding sites on the same protein? Do you think that's the key?
MAC1 has, for example, up to 120 repeats. One repeat is a 20-meric peptide, and this peptide contains 5 theoretical O-glycosylation positions. They are not all substituted, but most of them. So you can end up with hundreds of oligosaccharides, very complex, very heterogeneous, of course, but among these, also these blood group-like structures.
Thank you.
Other questions? Dr. Bode?
Just did a quick calculation. You said it was 20 to 50 millimolar, right? So that would be 10 to 15 gram per liter of oligosaccharides, which is exactly what we have in human milk. So total human milk oligosaccharides though, right?
Total, yes.
Right. So if we think of human milk not just being one individual component, but having glycoproteins, having glycolipids, having some antibodies, having all kinds of other defense mechanisms, I would argue that it's not one particular compound that has these effects, but the interplay of all of them together. So if you add these oligosaccharides, and maybe it's only in the 10 millimolar range that it's effective, or 5 millimolar range. So we're far away from the IC50, but together with all the other components, it might actually make sense.
But actually, we have no animal model, and we cannot test all these compounds, and so we have just these artificial systems, and we need to stick to these.
Sure. Yeah. But I think it's a good argument that milk is not just the sum of different components. It's an orchestra really of multiple different defense mechanisms that play together in the end.
But I'm not arguing totally against the idea that milk oligosaccharides could make the race. So, for example, 2-fucosyllactose is tolerated by an adult person up to 20 grams per day. This is enough. Yeah.
More questions to Dr. Hanisch? Yes, Dr. Ogra?
I really enjoyed your presentation. I just was wondering, what is so unique about the oyster in the context of norovirus? And do you see norovirus infection in any sea animals also?
No, so far not. Well, the oyster has one advantage. We know that those viruses or noroviruses which are affecting or accumulating in oyster are generally also infectious in humans. So this is known for a long time. So oyster came initially into our mind because this is a known trait. Whereas pig, for example, have other strains. So they have the G3 strains, and dogs have other strains. So can't use any of these because they are not infectious in humans.
Well, I was wondering, do you see norovirus see norovirus outbreaks on cruise ships, for example,
Yeah.
a lot.
Yeah.
So I was wondering, is there any particular relationship to there?
It could be. I agree.
Any other questions? If not, I suggest we take a coffee break. We were up admirably early. Natalia has an announcement before we go and have coffee.
Thank you very much, and I think it's very good that we have a little bit time, because for those who live today, it would be a good time to check out. Or we do the latest just at the beginning of the lunch. But as we have a time now, please do so. Thank you, and see you later.
We start again
At normal time.
second part of this morning session. And it's a pleasure for me to introduce a friend and colleague from Sweden, Professor Olle Hernell. Olle has done substantial research on mechanisms of lipid digestion and studies on lipid metabolism in infants. He's done a lot of clinical studies. He has received several awards, among them the Lenander Award and the ESPGHAN Distinguished Service Award. We will now move into another compartment of milk. Olle will talk about milk fat globules, effects on microbiome, metabolome, and infections in infants and children. Olle, please.
Thank you, Bo. Thank you for an introduction, and thank you, NNI, for the invitation. We have heard over the last 2 days, 2.5 days soon, that there are striking benefits with breastfeeding compared to formula feeding. And that is irrespective of the fact that there is a continuous development of infant formulas to mimic the composition of human milk, but even more important, to try to mimic the outcomes of the formula-fed infant and the breastfed infant. But still, there are differences in growth patterns. We know that even if you can do randomized clinical trials, blinded studies, and if you try to adjust for any differences in socioeconomic status and things like that, breastfed infants tend to have better cognitive achievements, even in well-developed countries. Fewer infections, we don't know and heard that there are different intestinal microbiota composition. Reduced risk for obesity, reduced risk for type 1 diabetes, and then there are many other differences that have been suggested and shown, but are still discussed whether they are true or not. So the question is whether the milk fat could have anything to do with these differences in outcomes. And the milk fat is to 98% triglycerides, hydrophobic lipids that are the energy substrate. 50% of the energy comes from the triglycerides. And the milk fat globule has this core of triglycerides and other hydrophobic lipids, a little of cholesterol esters, for instance. When the lipid droplet leaves the endoplasmic reticulum, it is enveloped by a single layer of phospholipids to stabilize the globule, otherwise it would just coalesce. And then it's transported to the top of the endothelial cell, and when it's extruded, it becomes enveloped by the apical part of the plasma membrane. So now it's enveloped by a triple layer of principally phospholipids and some cholesterol, but also a number, several hundred proteins have been shown to be components of the milk fat globule membrane. And the interesting thing is that while the fatty acid composition varies between human milk and, for instance, cow's milk, the membrane, there are some differences between human and bovine milk. For instance, there are a little higher concentration of sphingomyelin in the human milk than in the bovine milk fat globule membrane. But the proteins are very well conserved between the species, which tells you something. It must mean something. And you have in the membrane glycerophospholipids, sphingolipids, cholesterol, and glycosphingolipids. The problem here is that when human milk, or when the formulas are produced, the milk fraction has been discarded and never used. And that has been replaced by a blend of vegetable oils. And that also includes the milk fat global membrane that has never been used. There has been a focus on the fatty acid composition, and that has been adjusted to mimic the composition of human milk, and also the triglyceride structure. In human milk, there are around 20% of the fatty acids are palmitic acid, and 2/3 of the palmitic acid is esterified to the SN2 position of the triglyceride. And that has been supposed to have a significant effect, for instance, on fat absorption. Personally, I doubt that. Nevertheless, that has been also mimicked by structured triglycerides that can be added to the formula. But until recently, there has been very little focus on the MFGM. If you look at the components more closely, the phospholipids, the glycerophospholipids, the sphingomyelin, gangliosides, cerebrosides, cholesterol, and the components of these, sialic acid, choline, inositol, all these are important structures in the brain. And they are needed for brain development and for brain functions. And there are lots of data from animal studies and from in vitro studies that confirms that. If you look at the proteins, the mucins, butyrophilin, lactoaderin, glycoproteins, CD14, TLR1, TLR4, xanthine oxidase, and we have heard others mention them. So these, the membranes seems to be important. So the topic of this presentation was actually the effect on infections. And on the, if you look at the studies, you have this one is from Sweden, Peru, Belgium, India, China, France, and Italy. So many different countries, and you can see that the ages during the intervention has also varied. But let's start with the Swedish study. And that was actually the idea that our chairman and I had quite a few years ago now, when we were discussing together in Davis, what would be a reasonable next step if you want to try to close the gap or reduce the gap in outcomes between breastfed and formula-fed infants. And we came up with the idea that probably the milk fat global membrane. And we thought that if we could compare a formula supplemented with MFGM with a standard formula, that might actually affect the microbiome, metabolome, infection frequency, and also the cognitive development, at least some of these. So we decided to do that study, and we had a standard formula, a typical standard formula, and then the experimental formula. And as you can see, we had this experimental formula was reduced in energy from 66 to 60 kcal per 100 ml, also slightly reduced in protein to 1.2 grams per 100 ml, higher concentration of phospholipids and also cholesterol due to the addition of MFGM, this concentrate that was lacroprodan MFGM10 from Arla Foods. And the proteins constituted 4% of the total proteins in the formula. We included infants up to 2 months of age. You can't start to do a study in Sweden from day one, even if you want to do that, because virtually all kids are breastfed in the beginning. Then we randomized 160 infants to the experimental formula and the standard formula. And we had the intervention going on at 6 months, and the parents were asked not to start to introduce complementary food more than just a tiny bit, as the recommendation is in Sweden. And we followed disease symptoms until 12 months. And at 12 months, we also looked at the cognitive development with the Bailey test. And we had a reference group of breastfed infants that was also 80. Just to look quickly at the results, and this has been published, we didn't find any difference in growth between the two formula groups, and not between the formula groups and the breastfed group. And that is almost true. If you look at the weight and the anthropometrics at 12 months, there was no difference between the groups. But if you look at the growth rate, it was slightly higher in the formula-fed groups than in the breastfed group, but no difference in weight and length at 12 months. And no difference in percent fat mass between the groups, and surprisingly enough, no difference in protein energy intake, in spite of the fact that the experimental formula had lower energy and protein content. And the reason for that, the explanation, that was that the infants increased the volume. They compensated for the lower energy by increasing the volume, so the intake was the same in the two formula-fed groups. This shows the community structure or the fecal microbiota, and as expected, you can see this is without introduction of complementary food and with introduction of complementary food or after that. And this is at 4 and 6 months combined. And there is a difference, as expected, between the communities in the formula-fed groups and the breastfed group. The breastfed group also had larger heterogeneity, and the richness was slightly higher in the formula-fed groups. After introduction of complementary food, the differences started to decrease between the groups. And the differences between the formula-fed groups was modest. Some infants in the EF group had higher percentage of akkermansia, and that was particularly before a complementary food was introduced. And that's, I think, an interesting microbe because it is said to have anti-inflammatory effects, and it's also, I think, used more or less as a probiotic to reduce weight. And then they found at 12 months that Haemophilus was less abundant among the experimental formula-fed infants compared to the standard formula-fed infants. And that, of course, might have some impact on the risk for infections, although that's no causality, of course. So let's look at the fecal metabolome. Again, you can see that during the intervention, the breastfed group had more heterogeneous fecal metabolome profile than the formula-fed groups. And as for the microbiome on the previous slide, the differences between the experimental formula and standard formula groups were modest. At 12 months, you could actually not see any difference anymore. So this is a serum metabolome. And then it's starting to become more interesting, because here you can see that there is even perhaps more obvious difference between the breastfed and the formula-fed groups. And again, it's reduced when you introduce complementary food, so that is an important factor. And the metabolites, when you start to look at them of the breastfed infants, that was indicative of a fatty acid oxidation. We found acetate formate, while the metabolites of the formula-fed infants were indicative of a protein catabolism. So that's different. You found urea, essential amino acids, and amino acid catabolism byproducts like 2-hydroxybutyrate and 3-hydroxyisobutyrate. Now, if you look a little closer between the formula-fed and the experimental formula and the standard formula group, it tended that the experimental formula group was actually closer to the breastfed group that was indicative of fatty acid oxidation. I'll show you, I think, yeah. This is, as you can see, the fecal metabolites at 4 to 6 months again. And I should say that these data are from a subfraction of the group. There are 15 girls and 15 boys in each group, so 30 in each group. And here you can see that it's more common. This is the standard formula group. The concentrations here in the fecal metabolome is much higher when it comes to amino acids and essential amino acids and the byproducts from the catabolism of these. So these are products of the bacteria to a large extent, I would say. But if you look at what happens here, then you have the experimental formula, and you can't read it, but you find among them butyrate and tryptophan. And that's something that we learned yesterday that is quite interesting to find here. And then if you look again at the serum metabolome, then you have the experimental formula here, and you can see you have these products. You have above the first, you have fatty acids, linolenic acid, stearic acid, and you have 3-hydroxybutyrate, and you have myoinositol and lysophosphatidyl-choline, which is quite interesting. Also, I come back to that. But also, if you look on the top, you have tryptophan here. So if you look at acetone, which is another ketone body besides 3-hydroxybutyrate, you can see that in the breastfed group, the production of acetone is considerably higher, and also 3-hydroxybutyrate. And the MFGM group, the experimental formula group, that's actually right in between here. So more of a fatty acid oxidation profile like the breastfed group than the standard formula group. So ketogenesis is part of that, and that is typical for breastfed infants, that they actually produce ketones, so they are used as an energy substrate. And that's what you can see also in the MFGM group, even if it's not as striking as for breastfed infants. So what about the serum lipidome? This is at 4 and 12 months, and again, it's 30 in each group, and you have the green is the standard formula, and the blue is the experimental formula. And you can see there's a clear separation. Both at 4 months, there is a separation between the groups. And if you compare at 12 months, there is no difference between the groups. But if you compare what happens between 4 and 12 months, there is a difference in both groups. But here, the difference is no longer existing. If you look here at the erythrocyte lipidome at 6 months, and this is for the whole group, as you can see, again, there is a clear separation. And the separation was mostly due to sphingomyelin, phosphatidylethanolamine, and phosphatidylcholine species. And most of those that differed between the species were higher, as expected, in the experimental formula group. So if you summarize this, the serum metabolome of breastfed infants is indicative of fatty acid oxidation. You find the byproducts acetate formate, including ketogenesis, free fatty acids, 3-hydroxybutyrate, and acetone. If you look at the serum metabolome of formula-fed infants compared to breastfed infants, It's categorized by protein catabolism, degradation pathway metabolites, urea, essential amino acids, amino acid degradation byproducts. Most likely that could be partly due to higher protein concentration in the formulas than in breast milk, of course. Some metabolites, as I mentioned, were different between the experimental formula and the standard formula, even if many of them or most of them didn't reach statistical significance. There were differences. The experimental formula infants had higher levels of phospholipids and lyso-PC, as I mentioned, and tended to have higher levels of fatty acid products, choline, beta-inositol, and ketone bodies, and lower circulation levels of amino acids, suggesting that these infants were more metabolically similar to the breastfed infants, preference for fatty acid oxidation, including ketogenesis. So these energy substrate-driven metabolic profiles, they disappeared, as I mentioned, when you started to introduce complementary food, and at 12 months, you couldn't see the differences anymore. But it seems as, it might indicate that MFGM have a role in directing infant metabolism in the beginning. So, were there functional effects? When we start with cognitive development, even if that is not the topic of today's talk. But we did the daily test at 12 months. And we found, as you can see, you have the experimental formula in red. The green is the standard formula, and this is the breastfed reference group. There was a significant difference between the experimental formula and standard formula, which was in this case around 4 points. And that is pretty much in the same ball game as you find between formula-fed and breastfed infants in meta-analysis or larger comparisons. That doesn't mean that this is a whole explanation to the difference. That's not what I'm saying, but it happened to be in the same ball game. But there was, in this case, no difference between the experimental formula and the breastfed group. No difference in the motor development or verbal development. So the question is then, is there a likely explanation for this? Yes, I think that there might very well be. The higher intake of phospholipids, that was reflected in higher serum and red blood cell concentrations of PCs, phosphatidylcholines, phosphatidylethanolamines, sphingomyelins, and lysophospholipids. The lyso-PC is interesting, as I mentioned, because it is one of the most effective transporters of docosahexaenoic acid across the blood-brain barrier. Then we have choline, serine, myoinositol, and ketones in circulation, and they can all be transported through the blood-brain barrier and serve as important substrates to support optimal phospholipid synthesis for the rapidly growing brain and the newborn infant. Ketones are used, as we know, as energy substrate for the brain, but also as building blocks, for instance, for cholesterol, and possibly also can act as a signal substance. And the interesting thing with ketones is that the uptake into the brain seems to be directly correlated to the serum concentration. So, why effect on infections? I mentioned that there are many factors in human milk that has antimicrobial or immune modulating effects. Some of these are associated to or components of the MFGM. And I mentioned butyrophilin, mucins, lactoaderine, CD14, toll-like receptors, xanthine oxidase, gangliosides, oligosaccharides, glycoproteins, and possibly more. So, how about the infants and infections then? Well, we looked at that also, and we found that there were, what we looked at was infections leading to antibiotics or hospitalization use. And the antibiotics use, I can mention, was lower in the experimental formula group. But also we can see that the infections defined in this way was lower in the experimental formula group here than in the standard formula group. And we look even more close, we can see that the difference was mainly due to less otitis media infections in the experimental formula group. And that's not so surprising in a developed country that otitis is one of the most common infections in newborn infants or infants. The bacterial colonization of the upper airways has been shown to be important for the pathogenesis of otitis. It also been shown that not only nasopharyngeal cultures, but also cultures from saliva at onset of the otitis correlate to otopathogens. So that's a reason why we looked at the oral microbiota in saliva. And overall, there was not a huge difference. But when you look at it more closely, you could see that some strains were more common in the experimental formula group, as we have here. These are no difference here. And some were more common in the standard formula group, which we have here. And one of them we think are particularly interesting. And that's Moraxella cataralis, because that is one of the major otopathogens. And it's interesting that it is more common in the standard formula group than in the experimental formula group. We also looked at pneumococcal antibodies after vaccination. And we heard from Valerie that breastfed infants shouldn't have too strong immune response. So that could be interesting to look at. And if you compare here, you have the breastfed group, the standard formula group, and the experimental formula group. The response to these different antigens, you can see, is lower, generally lower in the breastfed group than in the standard formula group. And if you look at the experimental formula group, again, it's somewhere in between. It's lower in most cases. It's lower than it is in the standard formula group. And for 3 of them, the difference was significant. So that's, I think, an indication for immune modulation. So what about the other studies that had looked at infections and MFGM? This is the study that was done in Peru, and Bo was involved in that study as well. They looked at 550 Peruvian infants, 6 to 11 months old, and they were fed for 6 months a complementary food with the protein source being either MFGM-enriched protein fraction, and that was also lactoprodan, MFGM10 from Arla Foods, or skim milk proteins, both containing 1 RDA of micronutrients and vitamins. And what they found was that the global prevalence of diarrhea was a little lower in the MFGM group than in the control group, and that was significant. And particularly, they found that the frequency of bloody diarrhea was lower in the MFGM group. This study was done in India, and that was again a prospective 12-week double-blind randomized controlled trial, 450 infants, and they were 8 to 24 months. That was a study run in 3 different sites in India, in northern India, and they assessed acceptability and efficacy of a high ganglioside complex milk lipid that was 2 grams and 3 grams whole milk powder, and the complex milk lipids was from Fonterra, or the control group with 5 grams whole milk powder. And they looked for the prevention of rotavirus infection. And they didn't find any difference, even if you look at here, all case diarrhea, no differences between the group, and if you look at rotavirus, no difference between the groups. But the problem here was that this year it happened to be lower incidence of diarrhea or rotavirus than they had expected. So if they look back, it was underpowered for finding a difference. This study is the one that was done in Belgium. Again, this is older infants, 2.5 to 6-year-olds. And for 4 months, they were fed a daily intake of a formula, a chocolate-based formula with or without MFGM. And this was from Billinger in Belgium. And that was 9-fold enriched in phospholipids. And the data were collected from parental dairies. And the primary endpoints were days with fever, diarrhea, coughing, or constipation. And then they also had secondary endpoints. If you look at these results, you have them listed here. You have fever, diarrhea, constipation, coughing, doctor's visit, medication, school missed. Days they couldn't be at school. And the difference was only days of fever and short febrile periods, less than 3 days. That was the only significant different outcome between the groups. So we take the next study, and that was just recently published, a study that Bo and I were also involved in, and that was made in China, in Beijing, Shanghai, and Nanjing. And the idea was to evaluate the effect on growth, that was the primary outcome, and infections, rates of supplementing infant formula with one arm was probiotic Lactobacillus paracasi, F19, and the other was bovine milk fat globin membrane, and that was, again, Lacroprodan from ALA. And then there was a control group. And the design, a double-blind randomized controlled trial. We had 800 infants, 200 in each group. That's 3 groups of formula, and 200 infants in a reference group of breastfed infants. And the intervention was from 1 month, 28 days, 21 plus minus 7, until 4 months, and the infants were followed until 12 months. This shows the effect on infections, and to our surprise, we didn't find any difference between the groups during the intervention nor after the intervention. One thing to mention here, which is, I think, a pity, there is no, as you can see, you have fever, you have diarrhea, you have days with fever, other infections, episodes, but they never look for otitis. And I think that was, it's just happened that Chinese pediatricians don't do that. They don't use otoscope and look for otitis. And I think that had been interesting to see. But if you twist the data a little and look statistically, it was quite clear that the standard formula-fed infants, but not the MFGM and F19, they had significantly more fever episodes and days with fever, but no episodes of diarrhea. So there was a difference when you compare with the breastfed group. And interestingly enough, during the intervention, the MFGM group was not significantly different from the breastfed group in any of the primary outcomes. And you can try to figure out why. It's not that easy. Finally, this is a study that has not been published, but it's under publication. And that was also done in China. That was a randomized controlled trial, 2-week-old infants in Anhui, and they were randomly assigned to 1 or 2 staged formulas. We had one stage 1 formula up to 6 months and a stage 2 formula from 6 to 12 months. And the formulas were identical. You had a standard formula, 228 infants, and then you had the intervention formula where they had added a bovine lactoferrin and MFGM, and the MFGM again was lactoferrin MFGM10. And you can see that the dropouts are quite significant in this study. So they looked at the outcomes at 12 and 18 months, and the primary outcome was Bailey scales and toddler development, the BSID-3 at 12 months. And secondary outcomes was tolerance measures through 12 months, and then they looked at not specifically on infections, but they had infections as an adverse effect. And that I think was remembering also. They found an effect of cognitive development at 12 months. As you can see here, the difference between the groups is quite significant when it comes to cognitive development. They also found a difference in language and motor development, but no longer at 18 months. But then the dropout rate was very high at 18 months. If you look at the infections, if you look at diarrhea, significantly lower in the MFGM group than the control group. Here they didn't have a breastfed reference group. And if you look at respiratory events, you have a lower upper respiratory tract infections and lower cough. So that was an effect on infections as adverse effects. And this study was, that's the last one, that was a non-inferior study. And that was a randomized controlled trial that was done in France and in Italy. And they compared 3 different formulas. One was a protein-rich formula that supplemented with a protein-rich MFGM. That was again like Plan. And the lipid-rich MFGM, that was from Fonterra, and then a standard formula. And basically, they found no difference in growth or major safety concerns. And if you look at the infections here, you can see that there was no significant difference between the groups. But if you look at some of them, you have otitis, ear infection. Here, you can see that actually, if you just look at the number, it's lower than in the control formula, but that was not significant. What they found in this study was a higher frequency of eczema in the group that was fed the protein-rich MFGM here. And on the other hand, if you look at these 2 groups, there are very few infants. So I think you should be very careful about concluding anything from that. And that has not been repeated in any other study. So, to try to conclude, MFGM, if you start to ask, what is MFGM? Because it's really not very well defined. These are fractions that has been tried to be purified with different methods. And if you look at the studies that I have presented, people have used lipid-enriched, phospholipid-enriched, protein-enriched, ganglioside-enriched MFGM fractions. And different age groups and different intervention periods have been used. So the studies are not comparable. And even so, I think the studies have shown that MFGM is safe, and which is not surprising. I mean, it's a fraction from bovine milk. And that feeding infants MFGM reduces the risk of infections in most, but not in all studies, affects the plasma and erythrocyte membrane, lipidomes, and serum metabolome, which might be reflected in improved neurodevelopment. Modifies the oral microbiome, which may contribute to less risk of otitis media. It has a minor impact on the fecal microbiome, but drives the metabolic phenotype profile towards fatty acid oxidation, which is typical for breastfed infants. And studies, I think, with comparable, well-defined, as far as you can define it, MFGM fractions, intervention time, dose, and outcomes are really needed before we can draw firm conclusions, I think. So this is just to acknowledge people that have been involved in the Swedish study and in the Chinese study. One of the PIs there was Shaonan Li. Dr. Lundendal, you may have seen before. Niklas Timbe was a PhD student in the study, and Tove Grip, so Magnus Dumoulin in Umeå was involved. Ingegerd Johansson was responsible for the oral microbiota, Erik Dumoulin for the Bailey testing, Caroline Slapsky responsible for the metabolomics and microbiome analysis. So thank you very much.
With that, this paper is open for discussion. Valerie?
Yes.
Thank you. I just was surprised by the observation of higher antibodies upon vaccination for pneumococcus with formula versus breast milk or the other. So was it total antibodies or neutralizing antibodies that were addressed there? It was an
I
ELISA or
have
it
to
was
look that up. It was not done in our lab. I think they were neutralizing antibodies, but I have to check it.
So
It has been published, but I actually don't remember.
okay. Would you comment on that?
No, not really.
No.
You may.
Yeah, I find it very interesting because at the same time you find clinically protection with the breastfeeding and the formula that is enriched in the milk fat globule membrane. So, yeah, looks like too much antibody may not be good. I don't know. I find it's really surprising observation.
Do you find the results surprising?
Yeah, I do.
Oh, okay.
Yeah.
Sorry. Well, I don't know how surprising it is. We did another study with F19 several years ago, and but that was not a formula. That was actually complementary food between 6 and 12 months. And we also had an effect that those who had been breastfed had lower response than formulas.
Many studies don't have the power to look at clinical outcome, and they look at antibodies as a proxy for clinical outcome. But that may not be appropriate in early life. Maybe you don't need that much antibody in early life to have protection from disease.
Yeah, but I think, as far as I know, I mean, it's not unusual that you find, if you do it early in life, that you see a low response in infants, in breastfed infants and in formula infants. I think that's been seen also in other studies. Okay. Yes.
Yes, Dr. New?
I think that some of the studies you showed looked like they were very well done, randomized, prospective studies. But when it comes to certain studies where you have 3 interventions, and it's a non-inferiority trial, and you have 10 different outcomes, that bothers me.
I completely agree. But the reason why I showed this last study, that is a non-inferior study, is because they had actually looked at infections, but I agree completely.
Dr.
Yes.
Oftedal, and then that one.
Yes, I was interested in how you modeled the milk fat globule membrane intake by the infants, because obviously that's not a constant in the sense that fat content is changing over the course of milk consumption. And I would think that the pattern of milk consumption by the infant would affect its MFGM intake.
Yeah.
And if you could comment on that.
Yeah, well, that depends on the size of the milk fat globule, and that may vary. If you have larger droplets in total, if you have the same amount of fat, but larger droplets, you will have less membrane.
Right. Question back there.
Hi, thanks for the great talk. I have maybe a couple questions. One is a follow-up to the conclusion you had to begin with, saying MFGM hasn't been well characterized. Mm-hmm. I'm wondering, with the different studies you've shown, it's defined differently with the components that are enriched in potentially protein aspect of it. And can you speculate whether there's other ways where, not necessarily calling it a milk fat globulin membrane protein, but rather coming in with a whey protein fraction? And also, I'm assuming that when you have it in the formula, it's not the membrane itself per se, because probably it will be degraded in some way, so you're talking about just the components going in. And the second one is whether you've looked at the human milk MFGM and characterized that comparing to what a bovine MFGM looks like. Thanks.
That's what I mentioned in the beginning. There are differences between the bovine, as far as it's known, there are differences between the bovine and the human milk fat globule membranes. But if you turn it around, it's surprising that the protein composition of the membrane is so similar between breastfed breast milk and bovine milk, because it varies much less than, for instance, whey proteins and caseins. So it seems to be important that something is conserved in when it comes to the membrane proteins. The fat composition we know in the triglyceride core, that varies. We also know that molecular species of glycolipids, for instance, vary because glycosylation differs between bovine and human milk. When it comes to what happens if you add MFGM to an infant formula, of course it's not the same as if you have a human milk where you have the globule with the membrane. But no one has really, I think, studied carefully how the milk fat globule membrane is digested. And the only thing that I remember, and that we are talking about the 1980s, I think. Then there was a study that was done in Lund by Bengt Borgström's group, where they did electron microscopy. And what you could see was actually that during digestion, there were fractions of the membrane that was just shed off from the globules and was going freely into the small intestine and then digested. But exactly how it is digested, and if that does really have an impact on, so it means that there's a difference if you have just added the MFGM or it was actually in the beginning on the surface. No one really knows. Well, what is important is that if you look at human milk, the membrane has an important function because that affects the surface tension of the fat globule. And if you look at the digestion in vitro of a milk fat globule, then it's quite interesting to see that you have a specific surface tension. If you add gastric lipase, which is the first step in digesting the milk fat globule, you can see that if you have an emulsion particle and increases the surface tension, then gastric lipase has no effect, and then all of a sudden it starts to hydrolyze the triglycerides. And that's exactly at the surface tension you have in a milk fat globule. Yeah.
Yes, Dr. Embleton.
Thanks for a great talk, and I think we've all been very impressed by the studies. Could you speculate a little bit on the differences between kind of local effects and systemic effects? I mean, the most impressive outcome of the supplementation studies was a reduction in otitis media. And could you speculate a bit more? Is that due to some local modification of microbiota or lymphoid tissue, or is this more likely to be effect on systemic immunity?
No one knows, because we don't know whether there is one single component in the MFGM or if there are a mix of components that actually has this effect. This is a fraction that we have studied, and everyone else as well. So we don't really know what has caused the effect. The only thing that we have sort of tried to link is the higher frequency of Moraxella catarrhalis in the formula-fed group and in the standard formula-fed group. And that could be a link, but that's no causality, of course.
Dr. Ogra.
It was a wonderful talk. And as part of the question of otitis media, you also mentioned that the incidence of non-invasive disease was much higher as the otitis media went down. Is that right? The non-invasive bacterial disease went much higher in breastfed babies.
But they were higher, but the only thing that really came out as strongly significant was the difference in otitis media between the groups.
Okay, so that's the question I have, because, and with Moraxella catarrhalis, which we have also seen in the past, that one of the most important determinants of symptomatic otitis media is prior viral infection, like RSV and flu and other things, you know. In your studies, you have not looked at prior RSV infection or anything else, whether that could be an important determinant for the reduction in otitis media in these patients.
No, I can't answer that. We didn't look at that.
Okay.
Good point.
Any further questions? Otherwise, I would like to thank Ola very much for a very nice presentation. The next speaker is Professor Nicholas Embleton. He is a professor of neonatal medicine at Newcastle, and he's done substantial research in the area of neonatology, and we have invited him today to talk about a very, very ambitious clinical trial that he and colleagues has performed, and he will discuss with us clinical trials of lactoferrin in the newborn, effects on infection and the gut microbiome. Nick, please.
Thank you. I think the microphones need to be a little bit lower for me. Great. So thank you very much to Bo and Natalia and NNI for inviting me. So I'm going to talk a little bit about a large study that we were involved in running, looking at lactoferrin in preterm infants. I've got a few disclosures. I've had funding from government sources, but also charities. But most importantly, my main disclaimer is that I'm a clinician and I'm not a basic scientist. And the talk today is going to cover some aspects of preterm infant nutrition in general, some of the challenges we face, but particularly focusing on NEC and sepsis. So most of what I'm talking about is relevant to preterm babies. And then talk a bit about lactoferrin and why we chose to study it, the large trial that we did, and then touch on some of the mechanistic work that we're still kind of working through at the minute. And one of the kind of questions I want to pose is how difficult can it be to do a high-quality nutritional study? For us as clinicians, the theory is amazing over the last few days. But what we want to know is how much of this is going to change what we do when we go back to the NICU. And this is the feeling of pressure and anxiety that I've been feeling since I've been here. I came fairly kind of confident that I knew what I was talking about, but as I listened to several decades or centuries of research expertise, I've started to get increasingly worried, but was comforted by going to the Olympic Stadium the other day where Coubertin said, just look ahead, speak frankly, and act boldly. So that's what I'm going to try and do for the next 45 minutes or so. And here's my career CV. I've not won any awards, I haven't got any medals, I haven't got any honors in anything, but I have looked after about 2,000 babies less than a kilo. So that's my credential. So here we are. This is where Newcastle is in the north of England. We have 3 NICUs in a big population area of about 3.5 million. We're the largest unit. We accept all the babies in for complex surgery. All the fetal medicine cases and diaphragmatic hernias are born with us. So we have a very kind of complex mix of babies. It's a beautiful city built on the banks of the River Tyne with a lovely opera house. And it would be impossible for me to come here and give a talk without discussing Brexit. We do agree we would like to move from where we are, but our proposal is that we just move a little bit further south, where the weather is considerably better than it is in Newcastle most of the time. A little bit about the kind of research group. We're a kind of collaborative group of clinicians and scientists that have really kind of just evolved organically over the last sort of 10 years or so. A lot of our work has focused on the microbiome and more recently the metabolome, but we're also doing some viromic work. Chris Stewart, who's done a lot of our work, has developed an enteroid model in collaboration with Janet, who's in the audience here, where we retrieve some tissue at surgery. So babies who go for next surgery, we get permission to take a little bit of the resected tissue and grow the organoids from stem cells in the lab. And that hopefully will provide us with a very appropriate model for testing some of these interventions in the coming years. So say a lot of our work is really focused on neck, but we do have some long-term follow-up studies looking at the consequences of early life growth on later metabolic outcomes, insulin resistance, and these things. And importantly, we have the Great North Neonatal Biobank, of which Janet is the director. And in this, we have an amazing number of samples from probably over 1,000 babies now. Most of these are stool and urine samples, but we've already set up a system to retrieve salvaged blood. So when you take a blood sample and you send it to the lab and there's a little bit of EDTA or serum left over, the lab kindly put it in a freezer for us so we can go back, because of course looking after 500-gram babies, it's simply not possible to go around taking blood from these babies whenever you want. And in addition to all those samples, we also have some samples of breast milk and tracheal secretions, so that's really what we're trying to do. We've also been involved in setting up some pretty big studies over the last few years. The feeding trial, this was 2,800 babies. Hopefully that will be available in the next few weeks. It's coming out in the New England Journal. The lactoferrin trial Elfin that we'll talk about, our mechanistic study Magpie, which was nested within the Elfin trial. But we've also been involved in the trial of exclusive human milk diet funded by Prolacta. So just to kind of set the scene for those of you who are not neonatologists, just to kind of give you the perspective of how difficult it is to feed these babies and just, to show you what their nutrient requirements are. Anybody know who this guy is here? Any Colombians in the audience? This is Egan Bernal, who won the Tour de France this year. These guys cycle up and down mountains for 6 or 7 hours a day, and then they do it the next day and the next day, and they do this for 3 weeks at a time. And during this period, they consume somewhere in the order of 7,000 kilocalories a day, yet they still only weigh about 60 or 70 kilos. And just imagine how wonderful it would be to consume 7,000 calories a day of all this wonderful food, but still be as thin as that. So that's what he needs in terms of energy expenditure to cycle up and down mountains. Compared to that, our babies on the neonatal unit probably require an energy intake of around about 120 to 130 kilocalories a day. Therefore, our babies require 20 to 30% more energy than the most energy-consuming activity known to man. Quite incredible, really. And out of all of that energy expenditure in early life, about 2/3 of that is consumed by the brain. So quite easy to understand if we don't meet macronutrient intakes, why babies may turn out with longer-term problems. So that's energy. And then just to show you protein, so a 24, 25-weeker probably requires somewhere around about 4 grams a kilo a day. To show you what that would mean for you guys or for me, it would require eating about 15 beef burgers a day. I mean, can you imagine how appalling that would be? But that just shows you how incredibly high nutrient requirements are in preterm babies, and therefore the challenges that we face, both in terms of giving them enough parenteral nutrition, but also enteral nutrition, and how we do all that without them getting NEC, necrotizing enterocolitis, or sepsis. And of course, the most important outcome for us is long-term cognitive outcome. The worst outcome of a preterm baby is impaired neurodevelopmental function. And humans, as you know, are quite different from most other primates. Most primates have the rapid period of brain growth prior to term. So they're born relatively mature. Whereas for humans, we know that the rapid period of brain growth happens in the 3rd trimester and the first few months after term date. So that between about 24 weeks and 2 years of age, we acquire about 90% of our final adult brain volume. And when we're thinking about preterm babies, as I say, developmental, motor, cognitive outcome are clearly very important. But as I say, we're also interested in these aspects of metabolism in later life. And there are lots of kind of challenges we face, but here are the four of the kind of very key morbidities that are probably going to be affected by nutritional intakes in some levels, so retinopathy of prematurity, lung disease, but particularly necrotizing enterocolitis and sepsis, which is what we're focusing on in this talk today. Again, just to show you how far we've come, this is data from our unit over the last sort of 15, 20 years or so. And what you can see is that, of course, there's a lot of kind of bobbling around because we didn't have so many 23-week babies 20 years ago. But what you see is that at every gestational age over the last 15 years, we've got better. So survival at 23, 24, 25, 26, it's getting better. And just take a note here that in our unit at 23 weeks, our survival, and this means surviving to going home and being alive at 12 months of age, is now in the order of 60 or 70%. That's incredible compared to what we were maybe 20 years ago. And we could spend a lot of time talking about why we've got better. We actually haven't had any new medicines. We've got the same surfactant, the same antibiotics, the same steroids, yet we're doing better. And one of the key factors is an ability to work together, both the nursing team but also the physicians, and this shared combined experience and knowledge is really what's made a difference here rather than a single intervention. But when you think about these 20 or 30% of 23-weekers who don't survive, or the 24-weekers, we now know that after the first few days of life, the most common reason for a premature baby to die is due to NEC and sepsis. And both of these key diseases are clearly closely related to our nutritional and feeding practices. So over the years, what we've seen is that better nutrition undoubtedly has led to improved survival. And this is a problem of global relevance because, as you know, around about 20 million babies are born either low birth weight or premature every year. So this is relevant to every country and every continent. But as survival improves, we're faced with the challenges of looking after increasingly complex babies, babies who may have had NEC and had surgery and now have short gut. So this is a circular relationship. As we get better in nutrition, we need to learn and know more to look after the complexity that we face. We've heard a lot about breast milk, and as I say, I'm not talking about that specifically today, but clearly breast milk is the most cost-effective intervention in the whole of neonatology. However much we think probiotics, or we don't think probiotics work, breast milk from the mother will always be better than probiotics. There's a lot of evidence that it improves maturation of the brain, it improves maturation of the immune system, but most importantly, it results in better cognitive and metabolic outcomes over the entire life course. So we're not questioning the benefit of breast milk clearly is, as we've heard in lots of talks, the question really for us is, what can we learn from breast milk that may allow us to improve nutrition in these babies? And this is my kind of simplified scheme of nutrition that I've been kind of trying to sort of think and develop, I guess, over the last few years. Because for me, nutrition is more than nutrients. People get very bogged down in neonatal units about grams of protein or micrograms of this and that. But really the nutrients are just one part of what I consider the nutritional package. So the nutrients are clearly your macronutrients, your micronutrients, etc. But then, as we've heard in some great talks today and yesterday, the role of the functional components, the HMOs, the growth factors, the enzymes, but then also the interactions with the microbial environment and the gut microbiota that we're very interested in. And then I would have the fourth component as the kind of technical socio-behavioral aspects of feeding. Direct breastfeeding, the impact of tubes, sensory experiences, taste, and all these things. So these are the four components, I think, that we need to consider when we're talking about nutritional interventions. This is a picture of Harrison, who is a 23-week surviving baby. Very sadly, his brother died at a few weeks of age. I'm still in contact with his family and his mum. He's now 8 years old. He's an amazing little boy. As you can see here, mum was doing the right thing. Here he is only weighing about 1.5 kilos, and still he's actually trying to breastfeed before discharge. So Harrison's mum was completely committed to breastfeeding. She hated expressing. She said it made her feel like a cow, having to go off to that room 8 times a day and express. She didn't enjoy any of it. But she said she did it because she was convinced that it was best for her baby. So it's a very important message for us in terms of how we support and encourage mums. But I'll be honest that when I started doing nutrition research maybe 25 years ago now, and I was doing studies of different amounts of protein, I kind of just thought about formula as being something that was effectively fluid and nutrients. And you tip it into this long tube, which is the gut. I can see everybody in the front row looking disappointed that I had no idea of all the importance of mucosal immunology. But this is what I thought. I think you just tip it in there, and at the end you produce this waste product. And effectively that's sort of nutrition, isn't it? Just work out how much you're going to tip in and how much you're going to absorb. And of course, what we've all learned over the last 10 or 20 years is that it's much more complex than that. There are all these relationships with metabolites, but also interactions with the immune system, the endocrine system, and your microbes. And so we've had to shift our thinking from thinking of breast milk as a food to being a biochemical fluid that is allowing the mother in some way to perhaps signal to her baby. Let's put this quick slide in here just to remind all of us that although we think about the protein in breast milk as being the kind of casein, the whey fraction, there are lots of different casein components, but proteomic studies have now shown that even breast milk has peptides, that there's some digestion of protein in the gut. So we have proteases there, but then of course we have to have anti-proteases to stop breaking it all down. And I think this just reminds us that when we're talking about protein and the peptide fractions, to understand the complexity of protein digestion and really how little we know in preterm infants. And again, this is my kind of simplified schema. This is, protein digestion 101 for medical students or for a lot of us as neonatologists, because we don't get taught this very well. But preterm babies are very different. They tend not to have the low levels of acidity that you have in adults. And so although there are lots of proteases expressed on the brush border, and there are lots of peptidases in the bacteria, and then when you finally get to the bottom end, there are anaerobes that are going to ferment the bacteria. We know virtually nothing about this in preterm babies. So that when we come back to talking about protein and how to lactoferrin and how lactoferrin may be cleaved into lactoferricin, we really don't know what's happening in preterm babies very well. And so although we start off with these complex protein structures, bit by bit as you go down the tract, the protein is denatured and switched into to peptides and amino acids. But as I say, we don't know in preterm babies very much about this. So as I say, we understand there are different kind of protein fractions, caseins and wheys, and the whey compartment particularly contains lots of proteins, alpha-lactalbumin and lactoferrin. And we need to sort of stand back and be impressed by the complexity of human milk and how this whole protein whey-dominated milk, particularly in colostrum, provides this ideal balance of amino acids that optimize brain and metabolic outcomes. So these hydrolyzed proteins and peptides do occur naturally, but as I say, these key steps we know very little about, but we suspect that gut microbiota are going to be very important. And I don't need to tell all of you here in the audience how complex the gut microbiota are, the fact that we should really think of ourselves as a superorganism with all these bacteria being involved in lots of different metabolic and immune processes. But we need to understand that human development would be impossible without these microbes. So if we didn't have these microbes, we wouldn't be able to digest foods in the way we do. And as many of you know, these weigh 1.5 kilos if you add them all up. So again, we can all take some comfort that all that extra food we've eaten over the last 3 days, when you weigh yourself and you get home tomorrow, you can subtract 1.5 kilos because it's not really you. And we're here because we know that the gut is the inner tube of life, and that, as I say, is the focus of our research. There was a nice paper that came out a couple of years ago talking about human evolution in the context of a microbial world, as if this is some sort of struggle, like a kind of tug of war, where on the one hand we have to learn to manage the beneficial microbes, but then exclude the pathogens and the harmful microbes. And if you think about that, that kind of challenge is very acute in a preterm baby, where the baby has to work out which bacteria and which other antigens you should become tolerant to and which ones you should elicit an inflammatory response. And if you think about a 24-week baby who is supposed to be in utero swallowing amniotic fluid, how on earth does the preterm baby know that the bifidos are going to be the good guys and the E. coli are going to be the bad guys? He's had no memory, no teaching to do this. So very easy to understand why gut microbiome development in preterm babies is going to be so complex. And you will know that the pattern of bacteria change over the life course. So what is kind of optimal for newborn babies is very different to when we're older. Major impacts, as we heard from Sammy talking about when you reduce breastfeeding and how that interacts with your microbiome. So we've learned quite a lot about how this changes over the life course. But we really don't know what should be healthy in a preterm baby. And if you kind of extrapolated this graph backwards, what on earth should these patterns look like? Because we have no model of a healthy preterm baby. By definition, every preterm baby is unhealthy. They simply shouldn't be here. And nature didn't evolve to look after them. So trying to work out what microbial community structure is optimal in a preterm baby is incredibly difficult. So we've heard a lot about the interaction with mucosal immune system between your bacteria, the dendritic cells that reach out and sample these cells and prime and impact with your T regulatory cells, and then how this impacts with the systemic immune system. And when I come to talking about lactoferrin and we start putting some extra lactoferrin in the gut, just thinking about all the different ways in which that might act. So it may act as a food for certain bacteria. There's some data showing it may be probiotic for certain bifidos. It may prevent the growth of other bacteria, particularly those that rely on iron as a source because it binds iron. But there's also data to show that it interacts with the gut epithelium, interacts with T regulatory cell development. So lots of different mechanisms by which lactoferrin might be working in our babies. And then when I come on to talking about the clinical trials and thinking about sepsis, and in a lot of our babies, we suspect that the coag-negative staph infections that may be associated with catheters or on the skin, this lactoferrin somehow got to do something getting through all this system if we really believe that lactoferrin is going to stop skin infection in our babies. So this is my simplified schema for clinicians, just to try and sort of understand that we're talking about gut microbial communities. So these are communities of bacteria, and that there's this continual interaction with nutrients in the gut and all the metabolites that they're producing. And then there are lots and lots of steps before you then get to the stool. So whilst we do a lot of our work by sampling stool collected from a diaper, really all this interaction is happening way up higher in the gut. And we simply don't have methods for us to explore what's happening to the microbiome or the metabolome, particularly in the terminal ileum where most of the neck disease seems to occur. So by the time these bacteria or metabolites have got down here, they're likely to be very different. That doesn't mean that we can't use this, but we have to appreciate how restricted we are in studying where we are. So say, welcome to a far, far away place. This bit here is, in microbial terms, 1 million miles from what's happening in the ileum. So we know that there are lots of different dietary components that are likely to impact on gut microbial ecology, so particularly the HMOs, but probably just every nutrient that you study will have some impact on gut microbial communities. This is very different between breastfed and bottle-fed babies. But then in turn, all these microbes are producing other key compounds. We've heard a lot about short-chain fatty acids and how these may prime the immune system. We know that some bacteria produce amino acids that we then may absorb. Lots of bile acid synthesis is associated with gut microbes, production of choline and some key vitamins dependent on microbes, but also a massive array of anti- and pro-inflammatory mediators and signaling molecules. So this is unbelievably complex, and no way a humble clinician like me is going to unravel what is happening, particularly because we can't get to the bits that we need to get to. And we haven't talked a lot about it in the symposium this time round, but we're all starting to understand the importance of the gut-brain axis, this kind of bidirectional biochemical signaling pathway, lots of different sort of routes that connect the gut to the brain. And as I say, when we're thinking about maybe lactoferrin or some of these compounds, thinking about the peptides that may themselves have activity in the brain. I'm a little bit skeptical about how relevant this is in preterm babies, but clearly there's been a lot of work in adults linking bowel pattern, bowel habit to certain diseases. For example, people who develop Parkinson's disease turn out to have a history of constipation that was present years before they developed the symptoms of Parkinson's. So there's some really fascinating relationships to explore. And it's possibly true that in the preterm baby, some aspects of motility may be part of this pathway. But as I say, we really don't have much work on this at the minute. So this is the normal kind of process in healthy term babies. Birth seeds your microbes and genes, essential for all these components. You're supposed to come out covered in maternal secretions. So I like this phrase, birth seeds, breast milk feeds. That's what nature intended. You're supposed to do all these things. And then you're supposed to go home and get some extra seeding from the environment around you. But when you contrast that to a preterm baby, nothing about a preterm baby is anything like what it is for a term baby. Often born by C-section, they're put in clean plastic incubators. We wash and scrub and alcohol gel our hands. Just imagine what bacteria can survive that whole process to colonize the baby. Then we stick needles through their skin. We put bits of plastic that can all be colonized. We feed them with very abnormal regimes. Even the breast milk isn't produced in a normal way. It comes into contact with plastic and is stored before it hits the baby. Then if you're in a naughty unit, you give all these horrible H2 blockers, and then they get lots of antibiotics. So it comes as no surprise that the preterm microbiome is completely different to a term baby. So we've had this sort of diagram earlier before that there is this continual interaction between these three components, between the microbes, the nutrients, and the immune system. And what's optimal at the beginning of your life course is different from what's optimal at the end. And we can think of NEC in a very simplistic fashion as being a breakdown between these three components. So some it may be more immune-driven, some it might be more microbial, some it might be more nutrient. But somehow there's a breakdown between these different elements. And we talked a lot about evolution, and Olaf gave a great talk. I mean, it is absolutely fascinating to think, how breast milk evolved. And everyone says, and this Russian Dobzhansky. Nothing in biology makes any sense except in the context of evolution. But what about preterm babies? Because preterm babies have no pressure on evolutionary effects. And we heard from Joe that, we see these changes before babies get neck. We move into a more kind of dysbiotic looking pattern, although we're not really quite sure how to define the dysbiotic pattern. But what we don't know is, are the microbes causing the inflammation, or are the microbes just a consequence of the inflammation? Is it nutrients driving the inflammation which are driving the changes in microbiota? So we really can't unpick this, but we do know that there's a signal there in the microbiota to be had. And NEC, as I say, is the most important thing. And that's because NEC in childhood kills more children than all of childhood leukemia and lymphoma put together. So for a lot of people, this is just seen, this is just a problem that happens in preterm babies on a NICU. We don't need to invest time in it. But you're more likely as a child to have been a preterm baby who dies from NEC than all of these complex oncological problems. And we think about the major advances, thank God, and the treatment and success of these things. But when you think about NEC, really, our treatment hasn't changed in the last 20 years. We're no better at predicting it. We might be a little bit better at preventing it in some babies. But we're looking after more babies who survive. We're caring for increasingly vulnerable babies. Last year, we have our first surviving baby who weighed only about 350 grams. I'm not suggesting that's going to be a common occurrence, but somehow, almost magically, we managed to feed that baby without him getting NEC, and he's now a year old and quite healthy. So this is increasingly kind of complex for us. We're looking after lots of babies after surgery. And I'm going to be the third person to show an iceberg in the talk today, which is thinking that NEC is really just, for me, the tip of the iceberg of gut health. And lurking below it are all these other factors that relate to gut health and outcomes, duration of PN and antibiotics and cognition. So I like to think of a spectrum of gut health rather than a discrete yes, no, did he have NEC? I think if we just go for a yes, no approach, we kind of lose some of the subtlety in what we're talking about. And of course, it's possible that lactoferrin affects all these outcomes. So we use this horrible kind of histological term, necrotizing enterocolitis, really for a clinical syndrome. NEC is not a single disease. It's the end result of lots of different pathologies, lots of different kind of causative factors. We never understand the etiology in an individual case, and treatment is specific and crude. So here, this is a baby with black necrotic gut. He's definitely got NEC. Here's a completely healthy baby. At what point along that line do we choose to say this baby now has NEC? And if we can't be sure about where you are on that line, how on earth do we do studies to unpick it? So those of you who are clinicians in the audience, we can have great fun over lunch, arguing over which of these x-rays conclusively shows the baby has NEC, which one doesn't. If I told you that you have 1.4 mils of this colored aspirate, would that change your opinion? Or 0.5 of that, or really dark 0.5, but we love looking at these things on neonatal units. And then I'll show you a few bellies. Some are a bit distended. Some might have a little bit of umbilical flare. We put all that together, but this is how we make our diagnosis on a neonatal unit. It's really pretty crude, and we're obviously losing a lot of the signaling in all of this. And things are very different between units, so your approach to how readily you will stop feeds or start antibiotics or request an x-ray will change or impact on your instance. We know that there's variation in the rates of babies you get sent for surgery. So if your diagnosis is surgical NEC, there's a twofold difference between units and countries. How good is your histology? Do you really check this out? Are you absolutely sure that that was a perforation? Perhaps it was an isolated perforation where you get a secondary peritonitis and you miss it for 24 hours and then it looks like NEC. How do we work out whether this was a perforation or NEC to start off with? And if you don't review, or you don't have high post-mortem rates, or you don't independently review these things, how can you be sure really what we're talking about? So this is a massive challenge that we face. So we certainly need biomarkers. So what have we been up to in Newcastle? As I say, we've been working on this, particularly Chris Stewart has led a lot of these studies with Janet and myself over the last few years. I could show you lots of pretty pictures. I just want to sort of start off here because we found 12 twins and a triplet, and we looked at the stool microbiome. And what we were impressed by was how closely siblings clustered with each other. Maybe that's not surprising. But these babies are in different incubators. They're being touched by different nurses. Some are having antibiotics, some are not. But they're actually all quite similar. So presumably the breast milk exposures and maybe some genetic aspects really are quite important of this. So I'm going to come back to twins in a little bit. We were also able to look at the microbiome and show how closely the breast milk microbiome maps to the stool microbiome in some of these babies. So again, as we've said, we know that breast milk is a key driver of this. But what we've also been able to do is say with Janet's sort of painstaking collection of stool samples, if you want a stool sample from a preterm baby, go speak to Janet. She's literally got hundreds of thousands of them. But we could really look at the development over time. So here are twins where one twin gets neck and the other doesn't. So the red one doesn't get neck and the blue one does, and this is Shannon diversity. And you can see that maybe just a few days prior to neck, there is lower diversity as you might expect in the baby with neck. But if you go back a little bit earlier, there really wasn't any difference. So how on earth could we have predicted which of those was going to get neck? And we're then also able to look at the differences in taxa and show how things changed in the baby prior to neck, where you go, oh, look, increase in Klebsiella, that's not surprising. But then why didn't he get it here where he has more E. coli or something else? So it's just so difficult in individuals to predict what's going on. But also look here, these are where we put the antibiotics into the baby. Penangent and then Fluclox, Amoxigent and these things. These antibiotics don't have the big impact on the microbiome that you might expect. Clearly they're important, but we're not able to really manipulate the microbiome as much as we would think. So this was a great paper from Chris looking at 40 babies with lots of longitudinal samples, and I'm just going to show you how chaotic the microbiome is. So at the bottom, these dashed lines, these are the core OTUs. These are either Klebsiellas or E. colis or Enterococcuses, and the top of the satellite ones, these are the ones that are in much lower densities, things like bifidos. So here's one baby, just an example, just to kind of get your eye in. Over days 1, 2, 3, 4, 5, 6, 7, 8 of life. So he has Staph aureus, it disappears, then it comes back again. E. coli, everything in the baby at this point in time is E. coli, goes away again. Bifido, up, down. So completely random in this baby over a period of a few days. And then this is what it looks like in 40 babies. So just stand back and look at that and just look how different and how chaotic it is. How on earth are we going to unravel what's going on here? And baby 234 is this one here. 100% of everything in that baby's gut for 5 days apparently was Klebsiella. Like, I wouldn't like that to be my gut. But he was absolutely fine. So I think the key message from all this is that this is completely chaotic. And every baby is unique, and working out the disease one is impossible. So here's a heat map where Chris applied some clever statistics to break them down into different community types. And the computer model recommended that we would have 6 different community types. One is dominated by Klebsiella, 2 by Klebsiella enterococcus, etc., etc. Note that 6 is the only one with a substantial number of bifidos. And so each of these, I think there's about 800 samples here. Anyhow, so we have 6 community types. So if you hold those 6 community types in your head, this is what we did next. So we replaced each of those community types by a little dotted color for the next graph. And then this is what it looks like. These are all the control babies. These are the neck cases, and these are the sepsis cases. And you can see that each day we have a sample. We put them into one of those 6 different types. And what you see is that early on, maybe not surprisingly, there's a lot of staph dominating the infant gut. And then as time goes on, you start to see more bifido and more diverse appearances. But what we were able to see was that in the babies who got neck, they had many, many more transitions. They were moving between their community types much more frequently than the controls who appeared to be a bit more stable. So this was significant, of course, a whole lot of statistics there, so you can take it with a pinch of salt. But we do believe there's something about the chaos that's going on in those babies' gut that might be associated with neck. rather than just specific types of taxa. I don't know if they're paying attention. There's an animation here, if you can click on it. I haven't got a clicker. That shows a baby moving between the different community types. Can someone ask oh yeah, there should be a bar at the bottom where you can actually move the end. So yeah, so this is a baby moving around before he gets neck in red, and then coming in green is a baby who's healthy. And what you see in the baby with neck is that he's moving all over the place before he gets neck. Whereas the baby who stays healthy kind of moves to one kind of community type and then just kind of keeps around there. So that's a bit stylized because we obviously chose two examples that make the point. But I think there is something here about the kind of chaos that is associated with neck. So say neck's a major challenge. We've learned a lot, but we still don't know how to prevent it. Mechanistic understanding is really very limited. The animal models, as Joe was pointing out yesterday, I think are very, very limited for our babies. And of course, we have lots of ethical and practical challenges. But the fear of neck dominates decision making. And as I say, we should think of neck as being at one end of the spectrum. So the question is, does lactoferrin reduce it? All of you will know about lactoferrin, this important antimicrobial whey protein present in colostrum, breast milk, tears, saliva. Then the presence of acid is converted into lactoferricin, which again has antimicrobial, antiviral activity. Does this happen in a preterm baby in the same way? We don't know. We know that concentrations are particularly high in colostrum, and we know that all mammals express lactoferrin. And again, reading lots of the papers from Professor Oftedal and his group, understanding how these proteins existed before we were even mammals, that how you will find lactoferrin in worms is just amazing, how this protein has persisted. And there are hundreds, literally thousands of papers showing different activities and impacts of lactoferrin, which I'm not going to go through, but you can just take it that studies have shown impacts on microbes, viruses, fungi, lymphoid development. Lots of reasons this may be important in preterm babies. Lots of our babies get biofilms on their catheters, and lactoferrin may prevent this, prevents adhesion, disrupts virulence proteins. So lots and lots of theory that I'm not really going to get bogged down in today. So say we agree lactoferrin from your own mother is probably the best source, but these babies get really very little amounts of milk in the first few days. And a lot of mums don't provide sufficient milk. Human recombinant lactoferrin is available. There are some challenges with the studies, but it's likely to be very expensive. So what about using bovine lactoferrin? Widely available, probably pretty cheap. Similar in structure, but is it similar enough? So what did the Cochrane meta-analysis show on whether lactoferrin would be a good thing for preterm babies? So this came out a couple of years ago. These are all the trials in preterm babies, looking at the impact of lactoferrin on sepsis and neck. And what you see is that in 6 trials, you have a significant reduction in sepsis, and in 4 trials, a significant reduction in neck. So this looked like a very promising intervention for us to try. Just note also that this is in the wrong place here. So in the Sherman study, there was also a significant impact. So this is a sort of an error, printed or typo or something we need to correct. Anyhow, a lot of data there suggesting this could be a great intervention for our babies. So one of the challenges were all the trials were underpowered. I think there are issues about blinding and masking. And as we'll come on to, the source and quality of lactoferrin may differ between these trials. But we clearly felt we were justified in trying to put together a large, well-powered trial. If lactoferrin, that it was likely to be very safe, likely to be cheap, really does reduce neck or sepsis, even if it only reduces it by a small amount, what a wonderful intervention we could have. This is our unit. This is what a neonatal unit looks like. It is unbelievably complex. A lot of technology, a lot of equipment, very sick babies, very stressed parents. And trying to do clinical trials in this setting is incredibly challenging. And we're still learning. We've been doing these trials for 20 years now. But running trials in this environment is, as I say, incredibly challenging. So how do you do a good quality trial? Well, you need to make sure that you're asking a question that your users, your parents, public think is important. You've got to choose a great intervention. You've got to work out how much of the intervention to give and for how long and in which population. Then you've got to be really sure that you choose a robust outcome that's really meaningful. We wouldn't get funding to do a big trial like this without powering it at 90%. You can't do it at 80%. The funders just would tell you to go away at that price. And then when we went to get funding, we're competitive with the adults, doing huge trials of cardiovascular disease. So where did our trial start? Well, about 15 years ago, we put together a multidisciplinary group of neonatologists and dietitians. We formed in 2003. We meet 2 or 3 times a year. We're not aligned to any organization. We're completely independent. And this is our meeting in 2009. And I say, we're just clinicians. None of us had any academic time in our contracts. Here, the 6 of us here, we were the founder members from 2003. And then everybody else starts to come along when they see it's like a good idea. And Bill, who's sitting in the back here, had this great idea we should do a trial of something called lactoferrin. I had this amazing idea that we should do a trial of early or late feeding in neonates. And I was so wedded to that I really just thought it was important I did a trial that was called ELFIN. Bill came along and decided that he would actually take the lactoferrin on, and he stole the acronym from me to being enteral lactoferrin in neonates. So not only did he take my acronym, he decided to do the trial. John here decided to do this trial but call it something different. And I was left to literally pick up the shit after they'd chosen what they wanted to do. So the question was, does supplemental bovine lactoferrin in preterm infants less than 32 weeks at that dose compared to placebo, which is sucrose, reduce sepsis? And we had a very robust case definition. This is the timeline. This is just to show you scientists how hard it is to get one decent paper out. 2009, sounds like a good idea. Let's put an application in. The SIF guys went, yeah, this is a good idea as well. They put their application a bit later. We were working on both of these together. Then you're going to get funding. Then the first supplier of lactoferrin pulled out. Then we had trouble working out what placebo. Then we had to get approvals. Look, we're already into 2012 here. So obtaining the product and designing the trial was really complex. You'll see Janet here sitting there laughing at our ability to try and work out how to blind it, because some of these things here are lactoferrin and some of these things here are sugar. So I thought we'd play a little game now to see whether you think we got the blinding right. Here you can see us playing around with lots of stuff on a desk in our seminar room. So number 1 here, who thinks that's lactoferrin? Who thinks that's sugar? Hands up for lactoferrin.
It depends on the degree of saturation with iron.
Yeah, that's cheating. Okay, so this one was lactoferrin. This one here, lactoferrin or sugar? Recombinant lactoferrin. 1-1. This one here, it's got to be sugar, hasn't it? This one here. This one here. Lactoferrin. This one here.
Iron unsaturated lactoferrin.
No. Cocaine. And when people came in and saw us at this table with all these different powders and started taking photos, we were worried that whether Bill was going to turn into our Colombian friend here. Okay. There was no food dye licensed for use in preterm babies. So we went with using brown sugar in the end, because at least it was different from sort of white sugar. Lactoferrin is nothing like maltodextrin, which is a powder. So we tried really hard. We had a lot of quality control and assurance over our products, a lot of complexities with European law. We used a product from New Zealand. We had to import it in. The first time a big bag of white powder arrived, it was sent back because they weren't sure what it was. We obviously need to make sure it was lactoferrin, so we know it was definitely lactoferrin. But as we can discuss, like, we only know it's lactoferrin. We don't know about the bioactivity. We knew a lot about iron binding, so of course very important that we had a product that was not too iron saturated. A lot of quality control about how long that would be stable for, 12 months, 18 months. We bought it from New Zealand because they've not had any of these nasty viruses. We had to make sure it wasn't contaminated with metals. You're giving it to vulnerable babies. Then when we finally got the stuff, and of course if you shake to dissolve it, you realize it froths because it's a protein, whereas sugar doesn't froth. So then we realized that if we put a little bit of water with the lactoferrin plus 1 ml of the mother's milk, and then you shake it, both the placebo and the lactoferrin froth. You then got to set for 30 minutes whilst the bubble disappears. But the lactoferrin was still a little bit different coloured, not surprisingly. But we had these purple syringes that provide a little bit of masking as well. So this was our actual lactoferrin. We put it in these pots. We then put a stopper on the pot so that nobody could look inside and try and work out what was what. And then you connect a syringe to this popper. You make this up into batches, one each day of life, and you provide them to unit. Straightforward, problem solved, until you realize that every hospital, and we had 37 recruiting hospitals, has a different type of purple syringe, and they don't all connect to each other, and worse still, they don't all connect to the same NG tubes. So then somehow we had to sort of work around that problem. We then get inspected by the directorate to make sure that we're doing everything properly. Next problem was we were allowed to store it at room temperature, which is officially less than 25 centigrade. But if any of you have been on a neonatal unit, you regularly get over 25. So we had to monitor the temperatures and make sure that we could keep it and check it. And all the time, the clock's ticking. So here's where we published it. Here's where we started. 10 years of our life. So can you imagine the complexity in providing this to 134 different sites, developing algorithms, randomization programs? We were also setting up the feeding trial at the same time. So this was all going on at the same time. That's only just published now. And then almost 5,000 babies between those two trials. There was a little bit of crossover, about 200 babies in both. And then we try and set up to sample lots of stool and urine. So I didn't look anything like this 10 years ago. I had very blonde hair, no wrinkles. This has been a little bit wearing for all of us. So we recruited 2,200 babies. We have the primary outcome in 99.5%, very low incidence of adverse effects. But late onset sepsis was no different in controls to lactoferrin. And this is the chief investigator when he finds that result. I mean, we were absolutely gutted. We set out because we thought there would be something there. Of course, we had equipoise. That's why we did the trial. But we didn't set out to prove this doesn't work. Why would you do that? So we were disappointed. And everyone said, well, surely it made something better. Maybe some of these other, retinopathy or BPD. Nope, doesn't make any of that better. People go, oh, yeah, but do subgroups. It must work in the small babies or something like that. So this was pre-specified. Does it work in small babies or just those on breast milk or just those on formula milk? Nope. So we all sat around going, what on earth did we do wrong? And this was the principal investigator meeting once we all had the results. We spent a lot of time crying, working out what did we do wrong. But we told ourselves, look, maybe it's just true that it doesn't prevent sepsis. At least the lactoferrin we used in the trial we did doesn't work. And we're saying, of course, the lactoferrin works. It just doesn't work to answer this question. So why do we do it? Well, Manzoni had a high-quality trial, 470 babies, a 3-arm trial. These are always complex. They always give you concerns about power. But basically, lactoferrin, and a probiotic or placebo. They showed really quite a significant result, 5.9%, only 4.6% sepsis in the lactoferrin and LGG group compared to 17% in placebo. That's a massive effect. Reduction in sepsis-associated mortality. No babies died of sepsis who got lactoferrin compared to 4.8%. And then they did this add-on trial that has raised a few issues. Sherman did a trial published a couple of years back using tau lactoferrin, 120 babies. giving every 12 hours. They see less sepsis, 17% versus 33%. They also did some stool microbiota profiling. I'm not going to go into that detail, but it did show differences, lower Enterobacters and Klebsiellas. Sounds like a nice outcome. But this was only in 23 out of the 120 babies, so really just a really tiny number of babies. So why might we have seen things different? Obviously, difference in methodology, lactoferrin preparation. The babies are different. We gave more per dose than in the Manzoni trial. Interestingly, 5% of the infections in Manzoni were fungal ones, whereas we very rarely see fungal infections in the UK now because people use prophylaxis. A lot of our neck and sepsis occurs in the first 10 days or so. So really, how much lactoferrin will they have received in that time? What about for babies put nil by mouth? This is an intention to treat analysis. And if you really just have 5 days of an enteral protein, do we really think that that's going to exert an effect at a peripheral site like an IV, stopping that baby getting sepsis? So lots of other factors. Maybe lactoferrin needs osteopontin or some other cofactors for it really to work. Maybe the level of iron binding was a bit too high. Maybe there's some problems with transport and storage. We were convinced that we have very good high quality control. We have to pasteurize it. I've had one person say, you shouldn't have pasteurized it, but seriously, would you risk giving a bovine protein that wasn't pasteurized? That's not possible for our babies. So, of course, bioactivity is going to be key, and we simply don't have a method for knowing whether the lactoferrin we used really was the optimal product we could have used or not. We were restricted by what we could get hold of. Is it synergistic with probiotics? Well, a third of our babies did receive probiotics. No evidence of synergy. So maybe it's the New Zealand cows that are responsible for our failure to get it into a better journal. Okay, so I'm not going to talk about this in detail, but just to say we've used the ELFIN trial and the SIF trials, which is our MAGPIE study, mechanisms affecting the gut of preterm infants, where we collect the stool and the urine. We're doing some microbiome metabolomic work, very similar to what Ollie presented earlier on. And in our unit, we've been salvaging blood and doing some immune work as well. So we have 480 babies who are randomized in the lactoferrin trial. With comprehensive data, we know every day what these babies were getting, whether they were getting probiotics or antibiotics and what they were getting. We've got 30,000 samples from these, and we're currently, these are all going through the machines at the minute. So data will be available soon. And of course, we hope to sort of understand what lactoferrin was doing and why it may not have prevented NEC. And we're going to use this kind of twin design that I talked about, because what we found was that at least 100 of our babies in the trial were twins. So if you have twins that are both healthy, that neither one of them get NEC, what we did is we found, of course, that half of those twins will have got the same intervention. They'll both be on lactoferrin or they'll both be on placebo. And half will be discordant. One will have got lactoferrin, one will have got placebo. And to some extent, then we can additionally control for all the other things we've measured. Maybe we're controlling for some genetic factors here or breast milk exposures. And the babies who get disease, we're obviously looking at how the patterns change before they get disease. And we're using a match that is gestation, postnatal age, and hospital site matched if they haven't got a twin. Metabolomics, this has been a massive learning for all of us who are clinicians. This is really hard to get your head around and understand the new techniques as they come in. But we're doing a mixture of GCMS and LCMS. We're looking at stool, obviously, for products of microbial metabolism, but also looking at urine and plasma. Lots of challenges, but we're working with a great team who've got an extensive library of metabolites. And just to sort of say here, just in case it's true, but this is my prediction, I honestly think that there's going to be some really important signals in urine, and I can see how we could move to using urine as a sort of pattern recognition in our preterm babies, identifying the babies who maybe have some ongoing inflammation or high levels of protein breakdown or something like this. So urine's a great fluid. Stool is very problematic because the babies don't poop very much in the first few days, and if they get sick and have an ileus, they stop pooping completely, whereas the urine keeps coming. So I think urine could be a really important fluid that we need to explore. So these volatile organic compounds that you get on GCMS, we all have our own unique smell print. If you've got kids, you know which one is your kid and which one is somebody else's. This model's been applied to neck. Again, this could be a bedside technology. So here's a healthy twin, and here's a twin with neck, and you see a loss of one of these organic compounds. I'm not sure if they've identified it yet, but it's starting to suggest that you could maybe use this as a bedside technology. And it has already been used to detect rotavirus and C. difficile quite accurately in clinical settings. As I say, the group that we're part of is doing lots of other work, transcriptome and more histology, using stored fixed paraffin-embedded tissue from the lab. We can go back to the pathology archives, dig out all the samples with neck, and then do some work with these. We need better biomarkers. I'm not going to go through this, but obviously we're thinking about the stool component, the stool compartment, the urine compartment, and the blood compartment. And we're thinking about what are the different markers that maybe we could use to allow us to better predict. So these may be relevant at different stages of the neck disease in terms of preventing, predicting, but also importantly allowing us to subtype, classify neck in these babies, but also do more mechanistic studies. So hopefully that's going to come over the next few years. So summary lessons, clinical trials, nutrition, incredibly complex and very, very hard work. What works in the lab may not work in the NICU. Nutrients don't function in isolation. Maybe it's artificial just to pick one out and hope it's going to work. Observational microbiome studies are always confounded, sometimes very heavily confounded. At least we've done an RCT. Bovine lactoferrin isn't a human lactoferrin. Of course, we should focus on fresh breast milk, but as we said, we need better assays for bioactivity. But if you really want to know the answer in a clinical setting, you've got to collaborate, do high quality, i.e. large trials with functional outcomes, if we really want to move all of this amazing scientific knowledge to actually improving the health of the babies that we look after. So a massive thanks to everybody who's part of the team, Janet who's in the audience, and Chris who've done a lot of it. Chris Lamb in the lab, Greg Young, who looked after all our samples, our funders. Very importantly, all the babies and the parents who trusted us with their babies' lives to do these studies. So, as I say, good research is a massive team effort. I'm just presenting on behalf of some amazing collaborators. And I'm going to finish here. We've got a website for our group, neonatalresearch.net. We've got a tab here where we put all our slides. So if you want to go and read these slides or other slides, you can go and get it. And I'm going to finish there. So thank you very much.
Yes, Dr. Mestecchi first, and then Dr. Ogren.
First of all, lactoferrin is a glycoprotein. 10% molecular mass is contributed by unlinked carbohydrates, classical ones, and acetylglucosamine, fucose, and so on. But antibacterial activity of lactoferrin is strictly dependent on the presence of iron. Yeah. It can bind 2 atoms of iron, bivalent iron per molecule. And that's why you show the white versus brown. Yeah. If it's iron saturated, it's brown. And if you add EDTA and remove iron, it will be a white powder. So it depends. My question is, you obviously don't know the saturation with iron because only unsaturated lactoferrin is
I think the data is there. The company who provided us with this provided us with the level of iron saturation. I think it was 8% and it was stable.
Is it saturated or not?
Unsaturated. Only 8% was saturated is my recollection of the data.
So that have you discovered, have you detected any antibacterial activity? Lots of papers how to do that.
Yeah, we have all the stool microbiome samples, but we haven't got the data to share at the minute.
Best paper, Science, John Eldridge, 1980. Yeah. Gorgeous paper about degree of saturation and spectrum of bacteria with which it will.
So interact. I mean, we completely agree that the actual type of lactoferrin you use is going to be key to it. As I say, we did choose the product carefully. We had to choose a product that we would be allowed to use. We had to use something that was going to be available in sufficient quantities, that passed all the quality control regulations, and that was not heavily iron saturated. But you could argue, or we could argue all day about what would be the optimal formulation of lactoferrin to try.
If you mix it with whatever fluid which you give to the child per orally, if it has iron in it, you will saturate it again, and it will lose its activity.
We mixed it with water and then with breast milk, and mum's breast milk has very low levels of iron in it. So like that's the best that we could do. I mean, practically we have to find a way of getting it into the baby. So
Dr. Ogren.
Thank you so much. It was a wonderful talk. Jerry asked you part of the question, and I hope you are not disappointed. You didn't get any awards so far. But I'm sure you will get with number of patents you can have. But I just want to go back to lactoferrin itself. There are some very early studies with lactoferrin with secretory IgA, which I thought Jerry might ask you. In endogenous production in the mucosal tissues, you know. And have you looked at endogenous production in the respiratory tract or in the gut? And this may again be related to the saturation with iron. And that may be quite different, maybe functioning as effectively, maybe more.
Yeah, I mean, of course, we didn't set out to explore the role of endogenously produced lactoferrin. I mean, clearly that's very important.
Why not?
And all. Because I've got to look after a lot of babies as well, and I spend my time intubating, sticking drips in, and running clinical trials. I mean, if our whole research team was focused on lactoferrin, of course, this is where you would go. But as I say, at the end of the day, I'm a clinician, and my interest is in whether some of these ideas about HMOs, lactoferrins, choline, MFGM make a difference in the baby. And as I say, like, the theory is amazing. But at the end of the day, we have to take the step to decide whether you provide a baby at risk of whatever problem, cognitive, infective, whatever. You have to take the step to prove that some of this theory makes a difference. So that's why we chose to do this trial. But I agree, there's lots of other aspects of lactoferrin you could explore in these babies.
Thank you.
Yeah, Dr. Tlaskova.
It is understanding you correctly that in the resected tissue of neck patients, you do enteroids, you get enteroids?
Yes.
Because that's a phenomenal way to get information before you do your clinical trials.
Yeah, yeah. I mean, I think one of the challenges in neonatology is that there hasn't been enough basic science in the preterm babies. We still don't really understand the mechanisms leading to neck. So the enteroid system, I don't know how many we've got growing, but 20, 30, something like this are growing. So parents are very happy to support us. The surgeon doesn't resect any more tissue, but he just takes a piece of tissue that's not necrosed, and then Chris takes it off to the lab. So he has these enteroids growing, and as you say, that then provides us with a model, because these cell lines come from premature baby gut, and most often premature baby guts that are at risk of neck, because by definition they had neck. So we have a few controls for spontaneous intestinal perforations. We have a few later controls if a baby has like a stoma closure or something like that. So yeah, we hope that in the years to come these will be useful.
It's very expensive to do the clinical trials, and this gives you a lot of information about what your protocol is going to be.
Yeah, yeah, absolutely. We launched in clinical trial because 10 years ago, Manzoni wrote a paper that said, this stops babies dying of neck and this stops babies dying of sepsis, and we wanted to crack on and see whether that was true. So if we could do all this in parallel, we would. Sadly, I don't know what it's like in the rest of the world, but getting funding in the UK for this is almost impossible. We're basically full-time clinicians trying to do these trials. So.
Our last question from Dr. Tlaskalova.
It is supposed that very important factor for in complex neck pathogenesis is not well-functioning barrier function. What is known about the effect of lactoferrin in this direction, and are there some clinical approaches to improve this function?
Yeah, it's a great question. The answer is nothing. I mean, there is literally nothing on the mechanisms by which lactoferrin may work in these preterm babies. There are half a dozen small trials, and I know there's a lot of work in toddlers and infants and things like this, but there's nothing in preterm babies, and the physiology of the gut and the nutrient exposures and the microbiome and everything is so completely different in preterm babies that I don't know that we can extrapolate anything that happens in a healthy toddler to a preterm baby.
With that, I think we have to...
really important information. Lactoferrin is not only a free protein in milk and whatever, it is also within phagocytic cells, polymorphonucleoleucocytes, macrophages. So if you have inflammation in the tissue, you have plenty of lactoferrin, but it's not because of milk, but it's released by the cells.
With this, I think we have to finish the session. I would like to thank Nick again for a very nice presentation. And please notice that we only have 45 minutes for lunch today, and I would like you to please adhere to that, not only because I'm speaking, but we have a webcast scheduled to follow, and many people are departing later this afternoon. So please be back here by 1:15 or 13:15. Thank you.
Okay, thank you very much for being back. And we start the last part of the session today on the protective factors in human milk. And I have a great pleasure to introduce the next speaker, Professor Bo Lonnerdahl, Professor Emeritus of Nutrition and Internal Medicine, with multiple awards and multiple interests. But the key interest is infant nutrition, bioactive components in milk, and iron metabolism. And what he's going to talk about is effect of milk osteopontin on intestine, neurodevelopment, and immunity. Thank you very much, Bo.
Thank you very much, Natalia. I think I elevate these a little bit. I really appreciate being invited by NNI to share this with my distinguished co-chairs, Alan Walker and Ray Ogra. It's been a pleasure to work with them to get this program together. You heard about one single protein this morning. Nick Embleton talked about lactoferrin. I will talk about osteopontin, which I think is a little bit more new on the radar screen for most of you. And I will try to tell you a bit about osteopontin and what it possibly can do. Osteopontin is a very acidic phosphorylated glycoprotein and extensively post-translationally modified. There's about 28-30 different phosphate groups, and there are several glycans along the molecule. And more importantly, osteopontin contains an arginine glycine aspartate, or what is abbreviated for the amino acids RGD domain, which is actually an integrin binding site that works as a receptor for osteopontin, and also heparin binding domain, which is recognized by the CD44 receptor. So osteopontin has several receptors on the cell surface. It's synthesized by a variety of tissues, including the mammary gland, brain, bone, immune organs, and intestine. And as you can recognize, it was first localized in bone. That's because that's how it got its name, of course. It's present in most body fluids, such as milk, serum, and CSF. It's involved in a lot of biological functions: cell proliferation, cell survival, biomineralization, again coming back to bone, but many of them in immune function: leukocyte recruitment, inflammation, immune modulatory functions, wound healing, and initiation of cell-mediated immune responses. And I will show you some examples of this later on. We started analyzing osteopontin in breast milk. This is a collection of an NIH-sponsored breast milk study that I did some time ago, and we used that milk bank to look at osteopontin concentrations. And as you can see, it's very high the first couple of weeks, colostrum, but also in transitional milk, and then it maintains a stable but relatively high level up to 1 year of lactation. And it's actually the 5th or 6th most abundant human whey protein. So it's kind of surprising that you see so little of it in the literature. And it's very high. Like I said, this came out a few years ago from a Danish group of researchers showing that colostrum, again, human colostrum, is very high in osteopontin. Term milk is lower. But in particular, cow's milk is low in osteopontin. And not only that, but infant formula is even lower. During the processing, where you take skim milk and whey proteins and make it into infant formula, osteopontin is partially lost. So it's a very low concentration of osteopontin. This Danish group, they exposed osteopontin to gastric juice and found out that it seemed like it was resisting proteolysis. And later on, it was found that milk osteopontin can survive in vivo proteolysis in 20 days old mouse pups. And we have verified that, as you will see later on. A few years ago, bovine osteopontin became commercially available from Arla Foods in Denmark. and therefore the possibility existed to add it to infant formula. And why would you do that? Well, like I said, the amino acid composition of the bovine form of osteopontin and human is partially different. There's about 70% homology, but the phosphorylation regions are very similar. The glycosylation regions are similar, even if the individual monosaccharides vary a bit. But importantly, the integrin binding sites and the polyaspartic acid regions are totally conserved between human and bovine osteopontin. So both of them can bind to the same types of cellular receptors. We did a preliminary trial on this bovine osteopontin. This was done together with Sharon Donovan, one of my previous PhD students. And we fed infant formula with bovine osteopontin to infant rhesus monkeys. We believe that this is a very, very nice model for the human infant. Rhesus monkey milk composition is very similar to that of human milk. We can use it. They can get formula and feed themselves from basically when they're born, and they are weaned at about the same age as human infants, about 4 to 6 months. So this was a limited number of monkeys. As you can see, there were 4 breastfed ones, and then we had 6 each given formula without osteopontin and with osteopontin at the same concentration, roughly, as you have in breast milk. And in this case, which we unfortunately, of course, not can do in our studies on term infants, we got the jejunal messenger RNA, so we could do transcriptomics on the intestinal samples. There were no adverse effects at all. Growth and clinical parameters were similar. Breastfed monkeys were a little bit leaner, which they often are. And what we showed in the gene arrays was that there were 1,017 genes that were different between the breastfed and the formula-fed infants. But when bovine osteopontin was added to the formula, this diminished to 200. So it made them remarkably more similar to breastfed infants. And then there were, of course, some differences between the regular formula ones and those that were fed formula with bovine osteopontin. And we concluded from this one that it's safe. And of course, we were not too concerned about taking a regular dairy fraction and adding it to cow's milk formula. But we believed that the preliminary data from the infant monkey said that we could potentially have biological activities by adding this osteopontin. Because we saw these differences in gene expression profiles. The pathway analysis I don't show here, but the pathways mostly affected by the bovine osteopontin was those related to immune modulatory functions and to cell proliferation and differentiation. I then had the opportunity to do a clinical study where we added, similar to what we had done in the infant rhesus monkeys, we added bovine osteopontin to infant formula. This was done at Fudan University at Shanghai in China. Dr. Peng did a fantastic job on this study. It's a good site to do clinical studies, similar to what Ola and I have done in Umeå. Virtually all the infants there in Shanghai, in that area of the city, are breastfed from birth. About 80% are breastfed at 1 month of age. So that means that it's harder to find the formula-fed infants than it is to find the breastfed infants. But they have 10,000 births at that hospital, and therefore recruitment rate was quite decent. The formula was done by a Chinese company called Biostime, and Arla was providing the bovine osteopontin. In this case, we were fortunate because we were allowed to use 2 concentrations of osteopontin. As you can see, in very many of these clinical trials, only 1 dose level is chosen because these are work-intensive, they're expensive, and a lot of times you can just have 1 concentration. Osteopontin this far is relatively expensive to add to formula, and therefore it would be of interest to see if a lower level than what you see on an average in breast milk would have similar effects or not. So this is the breast milk average level. This is about half that level, but with the individual variation among women, there are women that will have this level in breast milk also. We had a regular infant formula without bovine osteopontin, and with a reference group of breastfed infants. And this was a randomized controlled trial, double-blind. And we did, of course, anthropometry. We measured it at birth and every month up to 6 months of age. We took venous blood at 1, 4, and 6 months of age. And the parents, together actually with the clinical staff, they visited them to make sure that the information was obtained properly. There was a feeding and dietary journal where they recorded formula intake. And also there was a very detailed morbidity journal that was supervised by the clinician also. We got tolerance data, stool characteristics, etc. We analyzed hematology, and we did flow cytometry at Fudan University. We got red blood cells that we sent to Sharon at Illinois, so we have done transcriptomics on those. That has not been published yet. We did serum ferritin at UCD, plasma amino acids, we did BUN, and we did an extensive battery of cytokines, also at UCD. And this one is a busy one. The bottom line here is I just want to show you that we had very low dropout rates. Ole showed some studies that have been done by other investigators where you have dropout rates of about 40–50%, and you'll start wondering how valid the data will be. We had less than 10% dropouts average after the 6-month observation period, which I think speaks really well for both the clinical investigator and her staff at Fudan University. Parents had 24/7 access to the physicians and nurses by telephone and home visits. We didn't see any significant differences in weight gain, nor did we see any difference in height. We also didn't have any significant difference in adverse effects. In fact, they were very low. There were no serious adverse effects, and the number of adverse effects was low and not significantly different. No difference in appetite and formula volume consumed, nor in sleeping time. I'm skipping a lot of the other clinical outcomes, but I would like to emphasize something here. And something that a lot of clinicians and nutritionists alike are not aware of is that infants fed formula have significantly higher TNF-alpha. In this case, it was not significant, but Erika Isolauri, who is at the same institute as Sameli Rautava in Turku, they have shown that formula-fed infants have higher circulating levels of TNF-alpha. And to me, that signifies that it's something we're not doing quite right with these. I think it's a signal that something may be going on down in the gut, which is a little bit out of whack. And if we could do something to normalize these levels, I think it is most likely going to be beneficial. Here you can see the TNF-alpha. This is confirming what you've seen in Turku. And you can see here that even if the baseline studies were taken at 1 month, when the mothers stopped breastfeeding and started with formula, they started with the respective formulas that they were assigned to by the randomized controlled design. And here you can see at 4 months, and this was exclusive formula feeding and exclusive breastfeeding up to 4 months. After that, and this was the intervention, after that they actually got some weaning foods. But you can see that with the repeated measures analysis, those infants that received bovine osteopontin at the higher dose here, they were significantly lower than those fed regular formula, and they were not significantly different from the breastfed ones. By 6 months, it was different, but like I said, at this time, they had stopped having the formula, and they were getting weaning foods, and there were no significant differences whatsoever. But it seems like it may be a dose response. We didn't see a significant difference here, but we saw it for the higher dose. We did several inflammatory cytokines, and the picture is similar. I will not show those. We also looked at anti-inflammatory cytokines, and the same thing happened there. The profile of cytokines became more similar to breastfed infants than those that were fed formula without osteopontin. Well, how does this translate into morbidity? Well, we registered days with fever, and here you can see, similar to what Ole showed for Sweden, it's a higher incidence of fever in those fed regular formula than in breastfed infants, and then adding bovine osteopontin at either of these two levels made them not significant from breastfed infants. So a reduction in days with fever. So it may be that the immune competence was altered to make them more similar to breastfed infants and more perhaps capable of fighting infections. We had subgroups of different types of infections. We did not have otitis media registered in this study, and we didn't see any individual group differences within each category. We also did very extensive, like I said, we did flow cytometry. Tina West, who is also from University of Umeå in Sweden, was in charge of that. And we looked at a lot of different immune classes. And what we found, to make this short, was that the group that got the high level of osteopontin had an increased proportion of T cells as compared to standard formula, and those that were fed half that level of osteopontin. We also had effects on the monocytes. There were other subgroups that were altered also. But we just concluded, and this was published 2 years ago, that osteopontin in infant formula at the higher dose level made the infants similar when it came to the distribution of different subclasses of immune cells, and therefore may affect the immune function. So to conclude from this clinical trial, and this to date is the only one that has been done in infants, more studies certainly need to be done. Addition of osteopontin resulted in no significant differences in adverse effects, formula intake, growth, or sleep, as compared to those fed regular formula. Immune cell distribution and cytokine responses were more similar between infants fed the higher level of osteopontin, the breast milk level, and the breastfed infants. Days with fever was higher in infants fed regular formula, but not in those that were given bovine osteopontin. And therefore, we concluded that feeding infants formula with added bovine osteopontin resulted in outcomes more similar to those of breastfed infants. With that, we moved on, and we actually analyzed osteopontin in the plasma of these infants. And here you can see that already at 1 month, there were apparent differences when it comes to human osteopontin in the 4 different groups. These are the breastfed infants, these are the infants fed bovine osteopontin at the 2 levels, and those were the infants fed regular formula. This became highly significant at 4 months of age, where they had been exclusively fed breast milk or regular formula or formula with bovine osteopontin. And this persisted up to 2 months after the intervention. And this made us to hypothesize, and you will see from the mouse that it's there's most likely autocrine upregulation of endogenous synthesis of human osteopontin caused by bovine osteopontin. And here you can see the analysis of bovine osteopontin in the serum of these, or the plasma of these infants. And you can see that at each sampling occasion, there was much higher levels of bovine osteopontin in the plasma when the formula contained bovine osteopontin. Note the scale. These levels of bovine osteopontin are considerably lower than those of human osteopontin, but it shows that certainly there was sampling of the bovine osteopontin, and we know that bovine osteopontin enters the enterocyte. We've done confocal microscopy. So some of the bovine osteopontin goes into the systemic circulation. Then we wanted to explore in the laboratory what the biological functions of milk osteopontin are, and we used a knockout mouse model, which I will explain in a few seconds. The reason we did that was that if you go in and do a PubMed on osteopontin, you will get a couple of thousand responses. It's in bone metabolism, it's in cancer, it's in just about anything. But if you go in and search for milk osteopontin, I think there is by now 5 publications. So we really don't know what the osteopontin presented to a newborn does to the newborn. We hypothesized from the literature that osteopontin may not only have effects on the immune system, but also on the brain and the development of the gut. And what we used here is, like I said, a knockout mouse model. And it's important here to recognize what we really did. We had wild-type mouse dams with wild-type pups. And then we had knockout dams that produces milk, which is totally void of osteopontin. And she fed normal wild-type mice. So they have the capacity to endogenously synthesize osteopontin in all tissues, just like normal, but they don't get that source of milk osteopontin. And then we did a whole battery of tests. What I will show you first is it's important when you have a knockout model that we didn't have an effect on milk yield, that they are lactating normally. We looked at the organ distribution in the pups by radiolabeling. And then we had these 2 groups that I just mentioned, those that were osteopontin negative, that didn't get osteopontin in the milk, and those that got osteopontin in the milk. We looked at intestinal morphology. We gave them an LPS challenge, and we looked at cytokine response to the challenge. We looked at normal immune function by cytokines, and we looked at the brains, and we also did a battery of cognitive tests to evaluate the brain function. And the first part of this was the brain. What does milk osteopontin do on the brain? It is abundantly expressed in the brain, and in rat brains it's been shown that osteopontin-expressing cells appear from embryonic day 16 and increase remarkably during the first week of postnatal life. And osteopontin has been shown to stimulate proliferation of neural progenitor cells isolated from adult rat brain. Again, very little have they looked at the newborn animal. And it's a well-known regulator of myelination, which of course is important for brain function and cognition and cognitive development. Here you can see, like I said, we showed that milk yield was identical in the wild type and the knockout dams. We confirmed, we killed sequentially some pups that were fed by wild-type dams, so they got osteopontin in the milk. And here is Western blotting using antibody against mouse osteopontin. And it's a little bit reverse here. You have the mouse way here with osteopontin at a couple of different molecular sizes. And here you can see in the stomach, it remains. Pepsin doesn't do a whole lot. And then you see some fragmentation to larger osteopontin fragments in the duodenum, the jejunum, and the ileum, so the first, second, and third part of the small intestine. So it does really survive throughout the small intestine in relatively large fragments, and these fragments do contain the receptor binding sites. We didn't see any significant effect on, on weight gain or body weight or brain weights in those that got milk osteopontin and not. We did histology. We didn't see any apparent differences in morphology between the animals. We looked at the expression of osteopontin in the brain, nothing significant, but when we did Western blotting of osteopontin in the brain, here you can see that we had highly significant differences. It's a little bit busy slide, but you have days 1, 2, 4 here, 6, 8, 10 here, and then you have those that got osteopontin in the milk, those that didn't get here matched. And you can see that there's a dramatic difference, particularly between days 6, 8, and perhaps also 10 between those that have been fed milk osteopontin and not. And then we went on, like I said, and did testing. And for those of you that are not familiar with this, this is the passive avoidance test, very popular to use. It tests more memory function than anything else. And you have a device here where the mouse pup will be put into the white compartment. And the natural tendency for the mouse pup there is to go into the dark compartment. They want to hide. Unfortunately, once they get in there, they get a small electrical shock, which they don't like, and they go out. And the natural tendency then for the normal mouse pup is that if you do that 1 or 2 times, you learn and you don't go in there to get shocked again. But those pups that didn't get osteopontin in the milk had a significantly poorer performance. They just couldn't remember as well that they got an electric shock there. So they tried again and they tried again. And this is showing that one part of cognitive function was impaired if you do not get milk osteopontin. We did another one which is a little bit more related to both memory, learning, and physical capacity. It's a rotarod test, and it's a rotating apparatus where the mouse is put on top of these rotating wheels. And again, if they learn, they are quite capable of kind of balancing that. They're rotating very slowly and successively going faster, and then the mice learn how to cope with that. But if you don't get milk osteopontin, you don't learn as well. Again, showing that cognitive development and memory functions seem to be impaired if you don't get osteopontin in your milk. Myelination, of course, is very important. You form the myelin sheath around the nerve to allow nerve impulses to move quickly. And it's a very complex process, and I'm not an expert on this at all, but it's really important for the development of proper function of particularly oligodendrocytes. And osteopontin is normally secreted by astrocytes and microglial cells and may stimulate proliferation of these precursor cells to promote myelination. And this is just a graphic picture showing some of these proteins that are involved in this. And you will see they are abbreviated here, but you have spelled it out in the next slide with the gene expression studies in the brain. And here you can see that several of the proteins that are involved here, cyclic nucleotide 3-phosphatidiesterase, myelin-associated glycoprotein, myelin oligodendrocyte glycoprotein, and in particular what many use to check this myelin basic protein. And you can see that these are normalized, so 1.0 here represent if you have normal in both the osteopontin plus and osteopontin minus mice. But here you can see that in the brain of the osteopontin negative ones that didn't get osteopontin, that the gene expression of virtually all of these is significantly reduced around day 6, 8, 10 as compared to normal. So very specific molecular events taking place in the, in the machinery involved in myelination, which may explain why we saw these functional deficits in the mouse pups. So milk osteopontin increases osteopontin expression in the brain. That's why I said it's most likely some type of autocrine regulation there. And the upregulated osteopontin expression may increase myelination and promote brain development. In retrospect, of course, it would have been very nice to have done Bailey test in the study we did in China, but at that time there was no suggestion even that milk osteopontin may have an effect on the newborn brain development. And the next study definitely should, in my opinion, have that included. Then we looked at immune development, and osteopontin is involved in the initiation of cell-mediated immune responses and promotes a Th1 cytokine picture and inhibits Th2 cytokines. It plays an important role in both acute and chronic inflammation, and it may exert both pro-inflammatory and anti-inflammatory actions depending on the nature of the biological scenario. This was from a review article from adult experimental animals. So what we did to mimic an infection, we gave LPS, which you have heard about earlier, so I won't go into that. And it's known that LPS can upregulate OPN expression in macrophages through TLR4 that we heard about yesterday. Not about osteopontin, but TLR4. And here you can see the results in the LPS-infected animals. And you can see here that interferon gamma, 4 hours after the LPS, was significantly lower in those that had not been fed osteopontin in their milk. And this was accentuated at 8 hours. And you can see the difference. We tested them at day 20, day 30, and day 40. And it was particularly pronounced here in the day 30 animals at 4 hours, and at the 8 hours in the day 20 and day 40 animals. TNF-alpha was the opposite, and there you had a significantly higher TNF-alpha response in those that had been fed osteopontin deficient milk. So a downregulation of TNF-alpha when osteopontin was in the milk, suggesting that pro-inflammatory cytokines may be downregulated by the milk osteopontin. So therefore, milk osteopontin may contribute to resistance to LPS administration and therefore bacterial infection by altering immune responses. More work needs to be done on that, and we're doing that as we speak. So, to conclude, I would like to say that milk osteopontin plays important roles in brain, immunological, and gut development during early life. Breast milk osteopontin likely provides beneficial bioactivities to the breastfed infant. And I think that is what we learned from the clinical trial. It suggests that if you would add something like bovine osteopontin that has similar binding sites as the breast milk osteopontin, you may be able to provide some of these bioactivities. And I think we need to explore much more what the osteopontin in the breast milk is doing to the breastfed infant, but that is more difficult to assess. Okay, with that, I would like to thank my collaborators, Dr. Jiang and Dr. Christine Prell, who did a postdoc with me from München. And with that, I'll end my presentation. questions? Yes, Dr. Ogra.
Bo, that was a wonderful presentation, really outstanding. Thank you. Because we don't know much about osteopontin at all. My question relates to the effects on the brain itself. Does the osteopontin which you give by mouth get into the central nervous system, or it stays in the gut and it interacts with the gut enteric nervous system?
No, we have done radio labeling and we can detect it in the brain, yes.
How does it get there?
That's a good question. We see it in serum, definitely. If it is then, and others have reported that it passes the gut, the blood-brain barrier.
I see.
If there are other routes to reach the brain, I don't know.
Well, I was wondering about the gut enteric nervous system, whether you produce a lot of, whether the osteopontin induces any neuropeptides in the gut, which may be stimulating the enteric nervous system via different pathways.
I wouldn't exclude that possibility for sure.
Have you looked at any neuropeptide synthesis in the gut?
No.
Serotonin or anything else? No?
We will do that, but we haven't done it. Thank you.
Okay.
Yes, Vanessa.
So on your bovine osteopontin trial, you showed one slide. Did it indicate there was endogenous osteopontin or induction of endogenous osteopontin production? Right. So have you thought about teasing out the effects of that osteopontin intervention as being directly related to the bovine osteopontin provided, or more directly related to the induction of endogenous production? So if you're looking at osteopontin-specific responses, do you think it has more to do with that the increased endogenous production in those mice receiving the bovine osteopontin? Does that make sense?
No.
It looked like there was a difference in the endogenous production in the mice that were provided the bovine.
Yes, that's correct.
So
when they got milk osteopontin, they got increased synthesis of brain osteopontin. Yes.
Right.
Yeah.
So
and in the human, we saw higher levels of circulating human osteopontin when they got bovine osteopontin.
I guess that's kind of my question, is perhaps the provision of the bovine osteopontin is maybe the effects you're seeing are more due to the stimulation of endogenous production of that osteopontin, and then the body's response to that, instead of the provided bovine protein itself.
In the human, you mean?
Yeah.
Yeah, that's possible. And that's why I said it's harder to tease it out in the human, where we have limited possibilities of chasing it. Yes, Dr. Oftedal.
Do you know how widely distributed osteopontin is across mammals? I mean,
no.
Don't know.
No, I don't know. I only know that it's found certainly in cow's milk. In mouse milk, it's similar to what we've seen for lactoferrin. Mouse is a high lactoferrin species, and it's also high osteopontin one. And the relative proportion of osteopontin in mouse milk is similar to human milk. We have, as of 2 weeks ago, we have found osteopontin in goat's milk, but we haven't looked at any other species.
And you said you did the monkey trial, didn't you?
We did, but we have not analyzed.
Have not looked at
no,
it. Okay.
No. But it's highly likely that so far all the milk proteins that are in rhesus monkey milk do cross-react with the human antibodies, and I would guess that the rhesus osteopontin, if it's there, which I think it is, would cross-react with the human one, so we could check that, yes.
Okay, and do you know whether it's found in other secretions?
Others have reported in other types of fluids, yes, but we have not looked at them.
Okay, thank you.
Thank you.
Did you look for or have any measurable amount of bovine osteopontin residual or innate into the regular formula as the control arm?
I didn't understand that.
The regular formula that was your control arm?
Yeah.
Was there any measurable innate osteopontin?
Yes, it was the concentration in that one was about 10 milligrams per liter as compared to the 65 and 130. Yeah.
Thank you.
Okay, thank you so much. With that, we'll go over to the last speaker of the day, which is Vanessa Dunn Castagna, who is a PhD student of myself and Dave Mills. So she has done a study looking at both milk components and the microbiota, and focusing in particular on the commensals. And I've been very impressed by Vanessa's progress in the lab, and therefore I discussed with my co-chairs, and we decided to invite her because I think it's a nice complement of what we heard the previous 2 days, but is focusing on a specific milk protein, secretory IgA in particular. So Vanessa, please.
Bring these back down. First, congratulations for making it this far. You're still here. I'm glad to see you. And much appreciation for my opportunity to be here to present to this amazing group. I've learned so much over the last 2 days, and I'm glad to be able to do the best I can to tie everything together. So I am focused on milk secretory IgA and its impact on the commensal microbiota. I will be talking to you about this complex here, the commensal secretory IgA interaction, specifically in how commensals, or at least the few that I have been analyzing, in its association with secretory IgA, the impact of this on commensal colonization, both in vitro and in vivo in a mouse model, and this investigation as to the ability of this complex secretory IgA and commensals to exclude pathogens. I don't have to go into much detail on secretory IgA. We have heard quite a bit over the last couple of days on this bioactive component. But just to remind you, this is found in abundance in both colostrum at about 5 mg per mil, and also over lactation age, which is seen in this chart, still maintains high concentration at about 1 milligram per milliliter with variation. This molecule is heavily glycosylated, and that's one of the things that I will focus on in this conversation. This is an image from Blaise Cordesi's review in Gut Microbes that does diagram nicely these N-linked glycans, heavily decorating the secretory component. The secretory component here, the heavy chain on both IgA1 and IgA2, and also extra O-linked glycans on secretory IgA1. These glycans have been analyzed quite extensively with Carlito Labria's glycobiology lab at UC Davis. This is one sample of this really amazing paper that looked at site-specific glycosylation and analyzed the relative abundance of these various N-linked glycans at each of these sites. This study was looking at 3 colostrum, so healthy women donating colostrum. And I did want to just kind of bring your attention to these sialic acid residues that we've heard discussed earlier today. And it looks like in colostrum that these glycoproteins are heavily sialylated, so with these sialic acid residues. And over lactation age, it does switch over to a more fucosylated, so more fucose residues on these molecules. So the function, and this is from a review by Nick Mantis's group from 2011 in mucosal immunology, that there are many, many functions already known of secretory IgA, and just to highlight a few from this nice review. We now know that not only are the secretory IgA molecules important in FAB-specific reactions, but these glycans also have a significant relevance in both immune surveillance and directly interacting with epithelial cells to reduce inflammatory response, including a reduction of the NF-kappa B pathway, an increase in the polymeric Ig receptor, which increases the amount of secretory IgA in the lumen, and even an increase in the tight junction binding proteins, so a promotion of barrier protection in these epithelial cells. We also know that with and without pathogens, so this is even with the presence of simply commensal bacteria, that we have a significant amount of sampling of luminal contents through a M cell receptors that are glycan specific, so requiring the glycans on the secretory IgA molecule. And this interaction allows this reverse transcytosis of complexes of luminal antigens, again, both with commensal bacteria and also with pathogens, to these invaginations where we have dendritic cells expressing DC sign, which is a lectin receptor, and the mannose residues on these glycoproteins that are involved in binding to these dendritic cells and leads to immune tolerance, so expression of interleukin 10. We also know that, again, this FAB-specific pathogen bindings, this is really classic immune exclusion or pathogen exclusion, known for decades about the function of this molecule with that specificity. But also, many studies have now come out recognizing the glycans as also relevant in pathogen exclusion of this molecule, including a study looking at the S-fimbriated E. coli inhibited by those sialic acid residues on these glycans. Clostridium toxin has been shown to be inhibited simply by the glycans on the secretory component alone, so not even requiring the protein itself. And then a number of other mucosal pathogens are also excluded due to the glycans. So a seemingly non-specific interaction with this secretory IgA molecule. And finally, numerous studies now have come out looking at the impact of secretory IgA on the regulation of commensal colonization. And again, this could be due to both glycan interactions, as Blaise Cortese has demonstrated back in 2011, but also now some new studies have looked at this low affinity interaction of secretory IgA with commensal bacteria. And this is really where my studies are focused. So what the gaps in knowledge that our research groups, both Dave Mills and Bo Lauderdale, we've been looking at are first looking at simply the interaction between secretory IgA. This is looking at milk secretory IgA, so a very heterogeneous group of this glycoprotein interaction with select commensals, and looking at its increase in colonization potential. So I've investigated the digestion of these immune complexes, secretory IgA commensal complexes, both in vitro and in vivo. The ability for this to improve attachment, both again in vitro systems and also in a mouse model, and then investigating immune tolerance in the host. And second, looking at the complex itself. So are there any specific functions to this commensal secretory IgA complex? And this I'm looking at colonization resistance, so potentially the ability for this complex to help reduce pathogen binding, perhaps improve pathogen clearance, and of course have improved barrier function on the host. And this knowledge that we are gaining with these studies not only will help us investigate mechanisms as to what's happening in a natural breast milk system, but potentially in the development of a product that may improve the ability for probiotics, or it may improve administration of probiotics. This is looking at a possibility of creating some complex, perhaps with Rhamnococcus, Lactobacillus, Bifidobacterium, some very common commensal bacteria of the infant, and creating a complex with secretory IgA and these bacteria that when administered to the infant may help protect the probiotic from digestion in the infant's intestine, and improve colonization once this complex reaches the infant's colon, and at the same time, perhaps prevent pathogen infection with that FAB specificity still with these secretory IgA molecules. That's a big goal, and I'll show you how far we've come. So I've chosen to use this list of commensals. I won't go into much detail on this, but these commensals up here, these are Bifidobacterium species. Some are subspecies, including B. infantis, which is B. longum subspecies infantis, B. longum subspecies longum. These guys up here I've chosen due to A, their ability to grow on specific human milk oligosaccharides. The two I've chosen to use in my study are 2-fucosylactose, or 2FL, and lacto-N-neotetraose, or LNNT. These guys up here, these BIFs, these bugs, have also been extensively studied by the Mills Lab. So we have extensive genomic characterization of all of these organisms. And we have a lot of really fun side projects on looking at their abilities to, or their gene expression alterations with the presence of their particular growth substrate and alterations in binding to this particular glycoprotein of interest. I've also included 3 different lactobacillus, or lactobacilli here. Lactobacillus rhamnosus 7469. This is the essentially the same species as an LGG. So I was looking at these as currently marketed probiotics, just kind of, again, looking at this concept of making this a translatable product. To obtain the secretory IgA, I just wanted to throw the slide up here in case you were curious. So this is the process of protein purification. I used donor milk for women who were over 3 months of lactation age and collectively pooled together numerous milk samples. With this, I created the skim milk from here and flowed this milk through a capture select IgA affinity chromatography matrix, which allowed for really nice purification. This is demonstration with an SDS page and Western blot showing a really clear isolation of this protein from milk with virtually no contamination. So with this pure product, I then was able to answer some of these questions initially, looking at the interaction between secretory IgA and commensals. First, is there a concentration-dependent binding of secretory IgA to commensal bacteria? And second, how important are these N-linked glycans? So does the removal of these glycans lead to a reduction in the association of secretory IgA? Now, if you're familiar with IgA and secretory IgA research, you'll know that Blaise Cortese has attempted or has answered the second question to an extent. And so part of this was recapitulating his studies, and the part of this was really a novel investigation. And so what I found, and I used flow cytometry to analyze much of my secretory IgA association. In this plot, you can see Cyto9 as my DNA chelator, and I used an antibody actually that Jeff Gordon used also in his study. This is a Alexa 645 detection antibody for secretory IgA. This is an example of a flow plot, and this is Bifidobacterium infantis, so B infantis that you're seeing here, where up at the top you can see an increase in my concentration of secretory IgA, and hopefully you can see a very clear demonstration of an increased association of my bacteria with secretory IgA with increased concentration. So certainly evidence that this interaction is concentration dependent. And I wanted to look at this a little bit more detail with some of the top binders. So Lactobacillus rhamnosus, that LGG guy, B. longum and B. longum infantis, all three had very robust binding under the highest physiological concentrations of secretory IgA. And when I used variations of this concentration, you can see that these organisms performed a little bit differently with their interactions with secretory IgA. But interestingly, all of them seemed to reach a saturation and then plateau. Interesting to me, and perhaps those of you in the breast milk world, because, again, the physiologically relevant concentration of secretory IgA in breast milk is about 1 mg per mil. So this perhaps demonstrates that in that breast milk environment, we actually have lowered saturation of our commensal bacteria that are found in that breast milk community than they could be reaching. But more investigation is required for this. To answer the second part of this question, how important are these N-linked glycans at this interaction, I utilized a glycosyl hydrolase that's derived from B. infantis. This is endo-BI1. This cleaves that chitobiose core. It does leave behind a small residue, but it efficiently does cleave these glycans from glycoproteins, and analyzed my results of these digestions with SDS page, demonstrating a clear reduction in glycan isoforms on here. And found with immunofluorescence that these D-glycosylated proteins indeed have a reduced binding. This is showing a picture of lactobacillus with a native secretory IgA molecule, and then here's lactobacillus, same concentrations with a endo-BI1 treated molecule, where you can see significant loss of association of secretory IgA, at least with immunofluorescence and this detection antibody. So certainly some demonstration that the association of secretory IgA can be reduced with the depletion of these N-linked glycans. With this information, I also wanted to look at this function. So what purpose is this interaction? And Blaise Cortese, in one of his early studies, showed that the secretory component alone was protective with the digestion of the IgA molecule. And so with this, I hypothesized that perhaps commensal bacteria coated with a secretory-coated IgA may also be protected from in vitro digestion. And so I developed a model. It kind of took a lot of trial and error to evaluate this question. So I passed bacteria 2 times. The second time would be on the sugar substrate of interest, glucose for all bacteria, lactose, and then the preferred HMO substrates. for some bifidobacteria. I checked the association, so I cultured these bacteria, bound it with secretory IgA, milk-derived, checked the association with FACScan, and then with this complex here, I then subjected this complex to gastric digestion conditions using pH of 4 with porcine pepsin, and then intestinal digestion with pancreatin at pH of 7. With this complex, I checked both live counting with CFU, serum dilution and CFU counting, and then I also used a flow cytometry with a cytotoxicity assay. And I evaluated this with all organisms of interest and found variations in the protective capacity of secretory IgA, but under all circumstances, with every bacteria analyzed, there did seem to be protective impact using secretory IgA. So this is log or fold change over these organisms digested without that protective antibody. In some cases, reached statistical significance, in other cases, very close to, but we'll be repeating some of these assays to truly determine this. But in all cases, if I did a regression plot using all different concentrations of secretory IgA, I certainly saw a demonstrated correlation behind the protective ability for secretory IgA to increase viability of bacteria post-digestion. So this was an in vitro model, and I wanted to see if this held true in a mouse model. And so in this first study, this is an investigation using BALB/c mice. This trial, we did a very short trial to investigate the survival, and I chose to use B. longum subspecies infantis. And again, this is one of these champion colonizers of the infant gut. So truly trying to tease out what's happening as best as I could with these infant commensals. For this trial, I collected baseline samples here to determine indeed that there were no B. infantis and really no bifidobacterium in these mice, followed by 3 days of gavage with B. infantis either associated or not associated with milk secretory IgA. I used that concentration that I had found was the highest saturation rate for this particular bacteria, and then collected fecal samples 1 day post-gavage, necropsied on the second day, to determine the amount of bacteria that essentially were able to survive that passage and persist for at least a short period of time. And what I found was indeed that when B. infantis was pre-associated with secretory IgA, and this was milk IgA, that I had almost a complete log-fold increase of bacteria that were able to passage through that intestinal content and be shed through the feces. Now, I will make one caveat that was one of my colleagues brought to my attention. This is fecal bacteria, so I cannot say with this plot alone that this demonstrates persistence in the intestine. We do have biogeography studies that certainly do show there is still almost the same, not quite, about 10 to the 5th CFU in almost every part of the intestine post-digestion, so at that day of necropsy. So I cannot completely say if this is due to just shedding of the bacteria or if it's evidence of survival of the bacteria. But nonetheless, again, with biogeography studies, I can say that these levels still did seem to be much higher in those mice that were provided that B. infantis that was pre-associated with IgA. So certainly, at least in this study here, we do see some evidence that secretory IgA can improve that survival of commensal bacteria post-digestion in these BALB/c mice. And the last part of this initial investigation with just commensals and secretory IgA was to look at this ability for these commensals to bind to both an in vitro co-culture system of human colonocytes and then also in BALB/c mice again. So for the in vitro trials, I, again, through much trial and error, came upon this concept of utilizing HT29-MTX cells, which have a very high goblet cell production, so I get a very significant mucosal layer that I think better represents what's happening in the colon. And I used Keiko2s also, so this is a co-culture. For these ones, I again used B. infantis, and I wanted to just do an evaluation of persisted B. infantis after a short period of time sitting on these colonocytes and look at any expression changes in the colonocytes themselves. And again, what I found was as I increased the association, and this is pre-association before I put the bacteria onto the surface of these colonocytes. So as I increased that percentage of population associated with B. infantis, I also saw a very clear increase in the amount of bacteria that were remain bound to the surface of these colonocytes, even after washing many times with PBS. And just to plot out, I took that slope and gave you a clear number. So for every 1% increase association for this bacterial population, you see about 10 to the fourth CFU increase binding capacity. So it seemed to be a very robust binding capacity with secretory IgA. And then again, gene expression studies on these co-cultures. When B. infantis was provided, and this is log fold or fold change over baseline with PBS alone, there was a significant alteration in a number of different genes involved in inflammation and also in tight junction binding proteins. And when secretory IgA was associated with B. infantis prior to that interaction with these co-cultures, essentially it brings things back to baseline. If you did look over B. infantis without and B. infantis with secretory IgA, you could say that there was increased tight junction binding, increased mucosal production, seemed to be beneficial for the introduction of this commensal to first have the secretory IgA association prior to that interaction with these colonocytes. And then again, moving this into an in vivo system, so looking at this inside of a mouse. This was again in BALB/c mice. You'll see a very similar timeline here where we wanted to collect baseline samples to ensure that there was no B. infantis in here initially. Gavage for 3 days with B. infantis. And here we have the beginning of my complex slide. So we have 4 different groups. 2 were provided water, but 2 were provided with 2-fucosyl-lactose to really increase the ability for that B. infantis to find its own little ecological niche and survive. We collected fecal samples day 4, day 6, day 8, and then necropsy on day 9 to look at persistence of B. infantis. And so what we found, I'll break this down just a little bit, when the mice were provided with water and B. infantis alone, there really was no detectable levels of B. infantis in that mouse on day 6, day 8. They were gone, washed out. When the mouse was provided B. infantis with that 2-fucosyl-lactose alone, we now were able to detect levels of persistence over all days of this trial. So in this case, it looks like that 2-fucosyl-lactose alone will increase persistence of this B. infantis. Now, this is also true if you've been following any of Dave Mill's work. We've also looked at this with B. longum longum, SC596, and found actually the same thing. So when these HMO-consuming bacteria are provided essentially their little suitcase of food, then seems to be that they can persist in even models where they normally would not. But of course, my question is, does secretory IgA do anything to this complex? So in this next group, this is B. infantis provided pre-associated with secretory IgA. And this is, again, back to that water model. So whereas we initially saw no persistence with B. infantis provided alone, now with just that provision of B. infantis complex with secretory IgA, you see them. They popped back up again. So it seems that secretory IgA did indeed increase the persistence of B. infantis, and this was over or almost to similar levels as just the provision of 2-fucosyl-lactose alone. So let's combine both of these together. What happens when we give these mice both 2-fucosyl-lactose and B. infantis associated with secretory IgA? It seems to be a combined effect. So with these mice provided both 2-fucosyl-lactose and pre-associated B. infantis, this spikes the levels up, and I didn't put my significance on here, but this group is definitely statistically greater than or higher prevalence than any of the other groups together. So a clear demonstration of the combined effect of both that specific HMO and secretory IgA. And again, just to kind of bring this back to what we're talking about in this whole group, right? This is the infant. That we're looking at something similar to an environment where the infants are exposed to commensal bacteria that perhaps are associated with secretory IgA, if you're looking at this in the breast milk environment, and of course have the presence of many different HMOs in that same environment. So perhaps this combined effect does really improve the ability for commensals to get into the infant and to colonize inside. So in summary for this first section, secretory IgA, I can say, certainly increases the colonization of select commensals. This was not true with every commensal evaluated, but certainly with B. infantis and a few of the others that we evaluated, including increased protection from digestion, improved colonic retention, and also improved host response. So with this information, I wanted to then look at the function of this complex in pathogen exclusion. We actually collaborated with the Wadsworth Center in New York with their model of Salmonella invasion. So we used Salmonella typhimurium, and we wanted to investigate essentially the functional cross-reactivity of a monoclonal recombinant secretory IgA called SAL4. This is specific to non-typhoidal Salmonella, and complexing this with B. infantis and looking at the prevention of this enteric pathogen, both in vitro system and also in these biopsy mice. So I'm just going to take a minute and explain this pretty cool model that we adopted from the Wadsworth Center. They generously provided these two strains of Salmonella typhimurium. One is the mutant strain, and one is a wild type. Both have kanamycin resistance, which allows us to grow these on selective agar, especially in a mouse system, so we can isolate these guys out. But the wild type has this O5 antigen that is specific to the SAL4 secretory IgA antibody. This has been extensively studied in the Mantis lab and also with Blaise Cortese, and shown to reduce the ability for this Salmonella typhimurium to invade and persist inside an animal model. And then this wild type has a deletion in a gene that's encoding the acetylation of the O5 antigen. So in this case, we have no SAL4 interaction, at least not a specific SAL4 interaction. I do have evidence that this still does bind to this guy, but does not seem to be due to that specificity of FAB. These guys also have that LAC-Z gene, a reporter gene that allows them to turn blue under blue-white screening agar. So again, a good way for us to differentiate one to the other. We chose, therefore, to use a competitive index, and this is both for our in vitro models and also in our mouse models, to evaluate the impact of the specificity of the SAL4 antibody. And for these competitive indices, we just did a wild type divided by mutant bacteria, and this is looking at those that were able to invade colonocytes or able to migrate to the Peyer's patches in a mouse model. And the idea here, a competitive index of 1 essentially means an equal number of both the wild type and the mutant. And a competitive index of anything less than 1 indicates SAL4 specificity, where we had a fewer number of the wild type and no change in the mutant. So we were looking at this as our baseline marker to evaluate the effectiveness of SAL4 specificity, and also to get away from the inherent variability in mouse models to look at Salmonella invasion. For our in vitro models, we used, again, actually for both, we used B. infantis. And for our treatment groups, we have 4 groups here. First is the Salmonella. This is our positive control, just Salmonella alone. Second is the Salmonella mixture. So we had equal molar concentrations, essentially, of the wild type and the mutant. And in this negative control, we have our SAL4 specific antibody that should be able to inhibit the wild type but not the mutant bacteria. And then our question mark, so B. infantis provided as a preconditioning to these colonocytes prior to Salmonella invasion or infection. And then finally, this truly a question mark here, does this complex of SAL4 with B. infantis have any specificity at preventing invasion of Salmonella? And what we found. First, just looking at total wild-type bacteria able to invade a colonocyte mixture. This is our co-culture system. This is our positive and negative control. So we do indeed see a reduction with that SAL4 specificity of our wild-type bacteria. When B. infantis was provided with no SAL4, we still saw levels that were the same as in our positive control, which was expected. And then now with B. infantis that was pre-treated or pre-associated with SAL4, we saw indeed, there was a reduction in the wild type, not exactly to the same levels, but still statistically significantly reduced for the wild type bacteria. And again, to measure the specificity of the SAL4 antibody to see if this really was that antibody that was doing this reduction bound to the surface of this commensal bacteria. To look at the competitive index, our positive control had no difference. This is just Salmonella provided alone. No difference in wild type versus mutant bacteria. When provided that SAL4 preconditioning, the wild type was inhibited and the mutant really was not. So we had a significant drop, about 50% less wild type bacteria than mutant. When B. infantis was provided as a pre-treatment here, we saw back to baseline, no difference in wild type versus mutant. And now the question of selectivity with that SAL4 antibody. Indeed, we saw that when B. infantis was pre-associated with that specific SAL4, the specificity of that antibody was still there, was able to selectively inhibit the wild-type bacteria over the mutant, indeed to even greater levels than when SAL4 was specifically provided to that complex. So this was a great indication that our hypothesis held true. B. infantis complex with secretory IgA was effective in reducing Salmonella invasion, and indeed was effective with that specificity. So the FAB specific binding was still present even when that B. infantis was associated with the secretory IgA. Looking at gene expression, this was just in the mammalian colonocytes. So we saw that B. infantis alone was able to reduce interleukin-8 production. This is a pro-inflammatory cytokine. This is Salmonella, and this is B. infantis. It did not seem to be secretory IgA had a significant impact here. But when secretory IgA was pre-associated with B. infantis, at least some tight junction binding proteins were increased with the presence of secretory IgA versus when it was not. And then finally, I'm almost done, finally looking at this, moving this into a mouse model. Again, the same kind of model here where we had 3 days of B. infantis gavage. And in this case, we wanted to look at the ability for that specific SAL4 response to Salmonella. This was our first control group where we had Salmonella provided alone with no B. infantis as our negative control, Salmonella provided with that SAL4 as our positive control. I'm kind of flipping those around. And then these mice were provided that B. infantis with or without SAL4 and infected with Salmonella 1 day or 3 days post B. infantis gavage. And again, we had different groups here, provided either water for these control groups here, or when B. infantis was provided, we gave these guys 2-fucosilactose to improve persistence of B. infantis. And what we found, first, just looking at total invaded Salmonella with both the wild type and the mutant, no difference. No difference in any of these groups with total Salmonella invaded into the Powers patch. But again, looking at competitive index was more our question. With the competitive index, when there's no B. infantis, just Salmonella alone, our competitive index was close to 1. And then when that Salmonella mixture was pre-associated with SAL4, we saw that competitive index drop as expected, showing you the specificity of that SAL4 antibody against the wild-type bacteria. So this was what we were comparing all of our treatment groups to see if anything showed the same level of specificity and reduction in invasion. Our first group, when we just infected with Salmonella 1 day post-gavage, there really was no difference from that initial competitive index. But here, when we had B. infantis that was pre-associated with SAL4 and we gavaged with Salmonella 2 days post B. infantis, we did actually see a reduction in the competitive index, indicating that this SAL4 that was pre-associated with B. infantis in the gut of a mouse 3 days after the presence of this or the introduction of this was able to selectively reduce the wild-type Salmonella bacteria from invading the Powers patch. So indeed, we did see the same or similar results as we saw in vitro, that this complex does indeed prevent that Salmonella invasion into the Powers patches, similar to the provision of that SAL4 alone. So again, this complex here, this idea of the function of this complex, we certainly can see that it was functional in preventing enteric invasion. And then overall, just looking at this, the concept of this function, increase the persistence of the commensal bacteria and perhaps prevent other enteropathogenic infections. And with that, I just wanted to say a great thank you to my enormous secretory IgA team that I've collected over time, and then both the Mills Lab and the Launerdal Lab where I do all of my work. And with that, I can open this up to any questions.
Thank you very much, Vanessa. Questions from Dr. Ogra?
Thank you so much. It's a wonderful study. And I promised myself I'm going to learn more here today. Can you define for me what you call commensal?
That's a good question. So we've chosen, and I use the term commensal most of the time in this conversation. So commensals are those that are derived from the infant, essentially these ones, the infant mammalian colonic tract, and able to essentially persist inside that organism. So these commensals, the ones that I've chosen here, were all isolated out of a healthy infant and used because we see them in the human population.
Because I don't think human body recognizes that this is a bad bug, this is a good bug. The bugs don't know they are bad or good. It
That's
depends
true.
on how they have adapted to the human host over
coevolution.
thousands of years.
Indeed,
really.
indeed.
So the question is the specificity of the system here. If you were to take a pathogen, what you call not a commensal, do the same thing, will it do the same thing as lactobacillus does?
So I've looked at this a little bit, and we looked at association with milk secretory IgA, with Salmonella, with EHEC, with Campylobacter, and do see that it can bind to these organisms, and it seems to be more preventative than it is increased persistence. So at least in the in vitro system that we're looking at, when I pre-associate Salmonella or pre-associate EHEC, for those, of course, it's a little bit different model. With Salmonella, I can see that actually milk secretory IgA alone is inhibitory to invasion of Salmonella to these colonocytes. And with EHEC, I see a reduction in adhesion of EHEC when pre-associated with secretory IgA derived from milk. So it does not seem to be doing the same thing. It does bind non-specifically, but...
See, that's what I'm a little concerned, whether this is a question of the model you allude. Because Salmonella is not a mucosal infection as such. It's a lymphoid cell infection. And it simply binds to the epithelium and then gets into bloodstream.
Yeah.
And the dose for Salmonella is pretty large and pretty variable, tremendously. If you were to use something like Shigella, would you be seeing something different? Or if you were to use a pneumococcus, which is coated with IgA, and give it instead of your commensals, would you see the same effect?
I don't know. I think that we should investigate that and see if we do see a similar effect. I mean, I did look at, as I said, 3 different enteric pathogens, and all seem to be holding true that milk secretory IgA still seems to be somewhat protective, even with Salmonella. Although you're absolutely right that the epithelial cells are not its end goal. It must still pass through those epithelial cells to get into their invaded tissue, and since that is actually what we're looking at, is just the prevention of that invasion.
But there's no specificity for the secretory IgA for the commensals which you used.
Yeah, it doesn't look
pretty
like
it. Any IgA probably could do it.
So how does it know is the question?
How does it know it?
Yeah.
You know?
That's a good question. I mean, and of course, we're looking at, in general, we're looking at gram-positive versus gram-negative here as well. All of the pathogens that we're looking at here are gram-negative, and the commensals, all of the ones we've chosen are gram-positive. And even Blaise Cortese, originally when he began this investigation, saw a difference and he used E. coli, the Nissl version, so this commensal E. coli, and saw that it did not perform the same as the gram-positive commensal bacteria. So perhaps the specificity is more due to just the gram nature of the bacteria and not necessarily to its pathogenic nature.
I think you did a fantastic job, the youngest and the last speaker for this conference.
Most of you stayed awake, thank you.
But I would really look at some of the quote unquote pathogens versus commensals.
Yeah.
Because they may function quite differently.
Yeah, they might.
And there are earlier data to suggest that IgA can bind meningococcus or gonococcus and prevent it from lysis by IgG and IgM also.
Yeah.
So there may be other mechanisms by which you can affect either uptake of the antigens or prevention of the antigen destruction by the host.
Yeah, one thing that I have not had the opportunity to do yet is do any mechanism-based investigation. I mean, the SALF4 antibody has been thoroughly investigated by the Nick Mantis' group and others, and looking at its ability to actually alter the bacterial cell itself, changing its flagellum motility, et cetera. And I actually found a reduction in even the wild type with SALF4. That specific antibody is still the mutant bacteria was still able to bind to that specific antibody and still reduce its invasion. So something else is at work besides that specific interaction with these antibodies. It'd be a very interesting study to do more mechanism-based research.
Thank you.
Yeah.
Dr. Walker?
Very nice presentation. I know that David Mill's favorite organism is Bifidobacteria infidus.
Yeah.
And I think he associates that with breast milk itself. So the question I have is, have you incubated that organism with breast milk to see if you get a complex that would occur in a manner similar to what you showed in vivo?
We have talked about that, but we've not done that yet. In part, no, I've not done that yet. I think I will. It has been done, just milk and commensal bacteria, and just pulling it out.
It has been done?
Yeah.
Because that would be a more physiologic approach.
Certainly.
Yeah.
But there are so many other factors in milk that could alter the binding of, BIFs to secretory IgA. There are different physiological functions or genetic alterations that happen with that bacteria in that milieu of milk. So it would be difficult. It would be an interesting just look to see what happens, but to figure out why it's happening, it would be a big question. And in part, not only is this study looking at what's happening in milk, but also what could happen if we took this out of that milk system and we're trying to introduce probiotics to maybe preterm infants, right? And making sure that we are not stimulating a robust inflammatory response with these guys, because obviously the infants don't know what's good and what's bad. So if we can protect these on the way in, it might be beneficial.
Yes, a question from Dr. Lifshitz.
Yeah, indeed, very nice presentation. My question goes a little bit to carry this type of finding to the human, and I would assume that the majority of the secretory IgA is produced in the small bowel. Is there evidence that the secretory IgA is alive and working in the human colon and not degraded by the microbiota?
Jeremy, no,
Actually, the density of cells producing IgA in humans in the colon is much greater than in a small intestine. So probably there is more IgA in secretions of the large intestine than a small intestine. So, why is that? I don't know, but probably because of the local stimulation, expansion of cells if you look at the distribution of bacteria in the gut, in the stomach you have basically nothing or maybe a few hundred bacteria, duodenum few hundred thousands, and you go all the way down and suddenly you reach the large intestine and you have to 10 to the 12 bacteria per milliliter or gram of the stool. That's a tremendous amount. And I don't know whether anybody, Ray, you may remember, co-culture or culture of intestine, small intestine versus large intestine. Tom Tomasi did something like that, but
Small intestine does produce more IgA, but there's a lot more space.
no, sure, because of the length, but per unity of I showed the slide of it from Per Brancek, there are more IgA producing cells per square millimeter than in a small intestine. There's no question about that.
But also,
But
IgA
it's,
is functional in the large intestine, regardless.
Sorry?
It is functional.
It's still functioning in the large intestine, even if it's produced in the small.
Oh, sure,
Yeah,
sure.
absolutely.
That's why milk may protect, because it passes through all the way.
Yeah, there's
Is that
certainly
answering your question?
There's absolutely evidence of bioactive secretory IgA in the colon. There are numerous studies we're looking at animal models with deficiency in secretory IgA or secretory molecules, and you see that what normally you see in the colon with it, which is the colonization of that outer mucosal layer, that those bacteria are able to really penetrate that outer mucosal layer and enter either into the inner or even to the colonic cells without secretory IgA or secretory IgM present. So certainly it is not only found, but most important in the colon.
One of the things I might have suggested to look at polymeric IgM also,
Yes.
especially in the absence of secretory IgA.
Absolutely.
And it may do as good a job.
It might.
Maybe better.
Yeah, I will.
Dr. New?
Have you given any thought to maybe coupling the SIGA to eco-groups, the types of things that Jeff Gordon has been studying when the Malawian children, where you take a consortium of bacteria and you take them from a set of healthy children, and those you then would add to malnourished children.
Yeah.
But if you could provide the most efficient route for that eco-group, it would make a lot of sense with what you're saying.
Yeah, absolutely. So we've not followed, we've not in our laboratory done the same levels of investigation as Chef Gordon does in identifying groups of bacteria associated with a particular phenotype. But the idea of creating a concoction, essentially, of different organisms has been something that we would really like to look at. And in particular, we looked at Lactobacillus and Bifidobacterium in part because it's theoretical that Lactobacillus are more small intestinal and Bifidobacterium bacterium are more colonic. So if we can create something that might essentially be beneficial all the way down the intestinal tract, especially if we're looking at prevention of enteric pathogens. So many are small intestinal, and BIFs might not have an impact on the small intestinal pathogens, but lactobacilli might. So it might be a really interesting investigation to take this concoction of both small intestinal and colonic bacteria, associate them all with secretory IgA, and sort of put them into a model with both small intestinal and colonic pathogens to see if it does the same thing. Yes, ma'am.
In case of IgE deficiency, IgA deficiency, if IgM is covering also similar
yeah, I think there was I did not do the study, but I think there was a paper looking at secretory IgM association, right? I forgot who did that analysis.
No, there are antibodies, yes, that has been done, but effect like you have examined, epithelial cells, that has not been done.
Ah, okay, yeah. But I do know, yeah, they do associate certainly with numerous members of the microbiota. Mm-hmm. Okay.
If no further questions, thank you very much, Vanessa. And with that, I would like to thank all speakers today, the audience that have been very diligent in following the whole day, and also I would like to thank the online participants that have been watching us today. And I'd like to also thank the Nestle staff that have been very supportive throughout the meeting. And do we have anything concluding remarks from Natalia?
It was a long 3 days, but they flew very fast. And I think the topic we discussed during 3 days about milk and mucosal immunity and the microbiome and how all this influence the neonate, it's really very important and on top of the interest of a lot of people. And Bo just mentioned and thanked people who were watching us around the globe, next to their computers, next to their smartphones. And we just got a number that during day 1 and day 2, we nearly got 2,500 people who were watching this program around. And day 3, we still don't know the numbers. And I'm sure it will go up because all presentations, video presentations, will be online till next Monday because people are working and we would like to give them possibility to watch these videos on demand. Just go to the Nestle Nutrition Institute website and log in and watch any presentation which has been presented during 3 days. And saying this, I really appreciate all your effort, and I hope that you will stay in touch with the Nestle Nutrition Institute, with the online platform we have, which is the Nestle Nutrition Institute website. You will share this with your universities, with your institutions, with your colleagues, because this is a very easy to stay updated on the different areas of nutrition and in different areas of research which are related to nutrition. And I'm sure that one day we will meet again, because human milk research is such an area which brings a lot of new findings every day. So I'm looking forward to meet you again. And with this, I would like to thank our 3 distinguished chairs, which is Pere Ogra, Alan Walker, and Bo Lonardal, who brought this program together. Let's give them an applause. And of course, without them, we would not have the faculty. So discussions which we had, maybe they were a little bit short because each speaker wanted to present too much information during the presentations, but discussions were very high temperature and everybody wanted to show their opinion. So thank you very much to all speakers who spent 3 days, and in 3 days they were trying to give us knowledge and expertise you have. Thank you very much. Our participants from different countries, very, very attentive participants from the morning till the evening. Nobody escaped for any, museum, any walk around the lake and any shopping. I really appreciate that. And I believe it's not only because of this, but because of the topic which we discussed. And you will bring this knowledge back to your countries, you will bring this knowledge back to your institutions, you share with your colleagues, and please use the Nestle Nutrition Institute platform, which will help you to share this information even further. Thank you very much. You got some publications during this workshop. We gave you just a little glimpse of what Nestle Nutrition Institute is doing. And if you go to the website, you will find all 90 plus books from the Nestle Nutrition Institute workshops. And next to this, another 3,000 of publications in area of nutrition, which are completely done for you and available for you anytime and anywhere. So thanks a lot. And the last one, but not the least, I really would like to say thank you to a few people who were all these 3 days in this room, maybe a little bit invisible, but without them, or maybe visible, but without them, this event would not take place. Perlita, thank you. The two ladies who are sitting very much in a nice environment, very calm, this is the NNI team. And actually, 4 of us, we are the NNI team who is doing all this work we bring to you, and we're really happy to share that with you. Thank you, girls. Thank you very much. And the people who are working with us for many years already. This is a clever team, technical team, which makes broadcast possible, video possible, and make it available for you on the online platforms. And Mael, I don't see you. Mael and team, thank you very much. So, saying this, I wish all of you wow, that's yes, 94th workshop came to the end. You still have time maybe to stay here in Switzerland and look a little bit around. For those who are traveling today, I wish you very safe and good travel back. And I'm absolutely sure we will stay in touch. Ciao. Thank you, both. It was a wonderful session. It was a wonderful session, yeah. So, when the next one?